and modify approaches. Success is a journey which necessitates smart work through learning, studying, and sacrificing. Despite all that, most of all, we love what we do.
The Faculty of Veterinary Medicine of Universitas Gadjah Mada was established in 1946. It was started from the Bogor Veterinary Medicine College. In 1947, a parallel class was opened in Klaten. Faculty of Veterinary Medicine has 4 programs: Veterinary Medicine Study Program, Veterinary Profession Education Study Program, Master of Veterinary Science Study Program, and Doctor of Veterinary Science Study Program. Various tasks and expertises of veterinarian are learned in 12 departments. Each department is equipped with advanced laboratories and modern research tools. The 12 departments are Department of Anatomy, Department of Biochemistry, Department of Pharmacology, Department of Physiology, Department of Surgery and Radiology, Department of Internal Medicine, Department of Veterinary Public Health, Department of Microbiology, Department of Parasitology, Department of Pathology, Department of Clinical Pathology, Department of Reproduction, Obstetrics and Gynecology. Besides an advanced laboratories inside all of the departments, this faculty is also supported with animal health education and training unit, used by university students and veterinarians for the sustainable learning activities. This unit is called UP2KH, which manage several livestock such as cows, sheep, goats, and horse. Various achievements have been obtained by the Solari Society, such as the Regan tester kit for chicken carcass. FVM lecturers discovered this tester kit. It has been patented under the name Durante and has been widely used by the community. Faculty of Veterinary Medicine lecturers regularly write and publish books related to the veterinary medicine world that can be used for readers in the animal husbandry, biology, and statistics. The university students has also obtained several achievements in the research, arts, and sports field, and also national and international organizations. Some collaboration in the national level has been made. FVM UGM is the member of Indonesia Veterinary School Association. An MOU has also been signed between the government and private sectors for the purpose of research and community service. As for the international level, FVM UGM has also collaborations with foreign universities from Australia, Thailand, and Japan for research and education and other abroad universities. Furthermore, FVM UGM accepts international students from abroad, such as from Malaysia. The commitment to develop science and community service is based on the veterinarian work principles, Manusia Mriga Satwa Sewaka. We are to serve mankind through animal kingdom.
Universitas Gadjah Mada or UGM is a public university renowned as the People's University, which strives for and puts forward the interests of the citizens and nation of Indonesia. As a higher education instituting, implementing, and leveraging education, research, and community services, which constitute the salient objectives of Indonesian higher education, or Tridharma, we remain committed to incessantly promoting the values of Pancasila. We exert the utmost effort to protect Indonesia's sovereignty in accordance with Pancasila ideology and the 1945 Constitution. As a world-class university, UGM is devoted to the interests of the nation and humanity. We conduct research, provide education, and deliver community services by enhancing knowledge that benefits the community, the nation, and the world. We have been learning from one another and get stronger together since we believe that knowledge is not useful unless it is shared with others. The learning process will never cease developing. At UGM, we learn by continuously exploring sciences, learning from history, observing the present, and researching for the future. We always work hard and never give up. The learning and research processes at UGM continue to run together and explore state-of-the-art innovations. To create a convenient, environmentally friendly, and pollution-free green campus, UGM has built a wisdom park that substantiates the teaching and learning process. The park per se is open to the public. UGM has also developed drinking water facilities throughout the campus, provided by Drinking Water Supporting Unit. This system is harnessed by the entire academic community of UGM. As the manifestation of its teaching endeavor, UGM operates various field laboratories for educational and research purposes. Furthermore, UGM has established and run a science technopark as an embodiment of its capability of inventing and commercializing a myriad of innovative health products, ranging from medical devices, herbal medicines, and healthy food.
There are now more than 118,000 cases in 114 countries, and 4,291 people have lost their lives.
2020 has been a tough year for anyone, and it is no exception for UGM. Campus activities are abruptly halted, student activities on campus are annulled, and everyone is forced to work, worship, and study from home. Lecturers have no choice but to learn how to teach effectively using the information and communication technology or ICT. Learning and research on technology are strongly supported at UGM, and in fact, we have conducted various cutting-edge technological research. Community Service or KKN is one of the most important courses to complete the study program, which will be undergone by University of Gadjah Mada or UGM in 2020 while adapting online courses system. Supported by the Ministry of Education, UGM will continue its service within the society across Indonesia during the pandemic issue. Lecturers and researchers at UGM continue carrying out basic and applied research in many realms. For instance, studies in the health sector to cope with COVID-19 are being encouraged and accelerated as a form of UGM's commitment to mitigating disaster using scientific methods. One of the most innovative inventions regarding to mitigating the COVID-19 is GeneNose, a screening tool or early detector of patients exposed to COVID-19, which is able to identify the symptom through exhale within 80 seconds. GeneNose is highly reliable. It has a number of advantages as a COVID-19 fast detection tool. Its sensor is able to detect more than thousands of patients in the long term, while providing relatively fast results, non-invasive and affordable. In addition, its usage also applies non-rebreathing mask and disposable HEPA filter. Those challenges somehow direct or even force us to innovate and modify approaches. Success is a journey which necessitates smart work through learning, studying, and sacrificing. Despite all that, most of all, we love what we do.
The Faculty of Veterinary Medicine of Universitas Gadjah Mada was established in 1946. It was started from the Bogor Veterinary Medicine College. In 1947, a parallel class was opened in Klaten. Faculty of Veterinary Medicine has 4 programs: Veterinary Medicine Study Program, Veterinary Profession Education Study Program, Master of Veterinary Science Study Program, and Doctor of Veterinary Science Study Program. Various tasks and expertises of veterinarian are learned in 12 departments. Each department is equipped with advanced laboratories and modern research tools. The 12 departments are Department of Anatomy, Department of Biochemistry, Department of Pharmacology, Department of Physiology, Department of Surgery and Radiology, Department of Internal Medicine, Department of Veterinary Public Health, Department of Microbiology, Department of Parasitology, Department of Pathology, Department of Clinical Pathology, Department of Reproduction, Obstetrics and Gynecology. Besides an advanced laboratories inside all of the departments, this faculty is also supported with animal health education and training unit, used by university students and veterinarians for the sustainable learning activities. This unit is called UP2KH, which manage several livestock such as cows, sheep, goats, and horse. Various achievements have been obtained by the Solari Society, such as the Regan Tester Kit for chicken carcass. FVM lecturers discovered this tester kit. It has been patented under the name Durante and has been widely used by the community. Faculty of Veterinary Medicine lecturers regularly write and publish books related to the veterinary medicine world that can be used for readers in the animal husbandry, biology, and statistics. The university students has also obtained several achievements in the research, arts, and sports field, and also national and international organizations. Some collaboration in the national level has been made. FVM UGM is the member of Indonesia Veterinary School Association. An MOU has also been signed between the government and private sectors for the purpose of research and community service. As for the international level, FVM UGM has also collaborations with foreign universities from Australia, Thailand and Japan for research and education and other abroad universities. Furthermore, FVM UGM accepts international students from abroad, such as from Malaysia. The commitment to develop science and community service is based on the veterinarian work principles Manusia Mriga Satwa Sewaka. We are to serve mankind through animal kingdom.
Universitas Gadjah Mada or UGM is a public university renowned as the People's University which strives for and puts forward the interests of the citizens and nation of Indonesia. As a higher education institution that has long been recognized by the society, we are proud of our timeless and tireless dedication to the nation. By cultivating, implementing, and leveraging education, research, and community services, which constitute the salient objectives of Indonesian higher education or Tridharma, we remain committed to incessantly promoting the values of Pancasila. We exert the utmost effort to protect Indonesia's sovereignty in accordance with Pancasila ideology and the 1945 Constitution. As a world-class university, UGM is devoted to the interests of the nation and humanity. We conduct research, provide education, and deliver community services by enhancing knowledge that benefits the community, the nation, and the world. We have been learning from one another and get stronger together since we believe that knowledge is not useful unless it is shared with others. The learning process will never cease developing. At UGM, we learn by continuously exploring sciences, learning from history, observing the present, and researching for the future. We always work hard and never give up. The learning and research processes at UGM continue to run together and explore state-of-the-art innovations. To create a convenient, environmentally friendly, and pollution-free green campus, UGM has built a wisdom park that substantiates the teaching and learning process. The park per se is open to the public. UGM has also developed drinking water facilities throughout the campus, provided by a drinking water supporting unit. This system is harnessed by the entire academic community of UGM. As the manifestation of its teaching endeavor, UGM operates various field laboratories for educational and research purposes. Furthermore, UGM has established and run a science technopark as an embodiment of its capability of inventing and commercializing a myriad of innovative health products, ranging from medical devices, herbal medicines, and healthy food.
There are now more than 118,000 cases in 114 countries, and 4,291 people have lost their lives.
2020 has been a tough year for anyone, and it is no exception for UGM. campus activities are abruptly halted, student activities on campus are annulled, and everyone is forced to work, worship, and study from home. Lecturers have no choice but to learn how to teach effectively using the Information and Communication Technology or ICT. Learning and research on technology are strongly supported at UGM, and in fact, we have conducted various cutting-edge technological research. Community Service or KKN is one of the most important courses to complete the study program, which will be undergone by University of Gadjah Mada or UGM in 2020 while adapting online courses system. Supported by the Ministry of Education, UGM will continue its service within the society across Indonesia during the pandemic issue. Lecturers and researchers at UGM continue carrying out basic and applied research in many realms. For instance, studies in the health sector to cope with COVID-19 are being encouraged and accelerated as a form of UGM's commitment to mitigating disaster using scientific methods. One of the most innovative inventions regarding to mitigating the COVID-19 is Genos, a screening tool or early detector of patients exposed to COVID-19, which is able to identify the symptom through exhale within 80 seconds. Genos is highly reliable. It has a number of advantages as a COVID-19 fast detection tool. Its sensor is able to detect more than thousands of patients in the long term, while providing relatively fast results, non-invasive and affordable. In addition, its usage also applies non-rebreathing mask and disposable HEPA filter. Those challenges somehow direct or even force us to innovate and modify approaches. Success is a journey which necessitates smart work through learning, studying, and sacrificing. Despite all that, most of all, we love what we do.
The Faculty of Veterinary Medicine of Universitas Gadjah Mada was established in 1946. It was started from the Bogor Veterinary Medicine College. In 1947, a parallel class was opened in Klaten. Faculty of Veterinary Medicine has 4 programs: Veterinary Medicine Study Program, Veterinary Profession Education Study Program, Master of Veterinary Science Study Program, and Doctor of Veterinary Science Study Program. Various tasks and expertises of veterinarian are learned in 12 departments. Each department is equipped with advanced laboratories and modern research tools. The 12 departments are Department of Anatomy, Department of Biochemistry, Department of Pharmacology, Department of Physiology, Department of Surgery and Radiology, Department of Internal Medicine, Department of Veterinary Public Health, Department of Microbiology, Department of Parasitology, Department of Pathology, Department of Clinical Pathology, Department of Reproduction, Obstetrics and Gynecology, Besides an advanced laboratories inside all of the departments, this faculty is also supported with animal health education and training unit, used by university students and veterinarians for the sustainable learning activities. This unit is called UP2KH, which manage several livestock such as cows, sheep, goats, and horse. Various achievements have been obtained by the Solari Society, such as the Regan Tester Kit for chicken carcass. FVM lecturers discovered this tester kit. It has been patented under the name Durante and has been widely used by the community. Faculty of Veterinary Medicine lecturers regularly write and publish books related to the veterinary medicine world that can be used for readers in the animal husbandry, biology, and statistics. The university students has also obtained several achievements in the research, arts, and sports field, and also national and international organizations. Some collaboration in the national level has been made. FVM UGM is the member of Indonesia Veterinary School Association. An MOU has also been signed between the government and private sectors for the purpose of research and community service. As for the international level, FVM UGM has also collaborations with foreign universities from Australia, Thailand, and Japan for research and education and other abroad universities. Furthermore, FVM UGM accepts international students from abroad, such as from Malaysia. The commitment to develop science and community service is based on the veterinarian work principles, Manusia Mriga Satwa Sewaka. We are to serve mankind through animal kingdom.
Universitas Gadjah Mada or UGM is a public university renowned as the People's University, which strives for and puts forward the interests of the citizens and nation of Indonesia. As a higher education institution that has long been recognized by the society, we are proud of our timeless and tireless dedication to the nation. By cultivating, implementing, and leveraging education, research, and community services, which constitute the salient objectives of Indonesian higher education, or Tridharma, we remain committed to incessantly promoting the values of Pancasila. We exert the utmost effort to protect Indonesia's sovereignty in accordance with Pancasila ideology and the 1945 Constitution. As a world-class university, UGM is devoted to the interests of the nation and humanity. We conduct research, provide education, and deliver community services by enhancing knowledge that benefits the community, the nation, and the world. We have been learning from one another and get stronger together since we believe that knowledge is not useful unless it is shared with others. The learning process will never cease developing. At UGM, we learn by continuously exploring sciences, learning from history, observing the present, and researching for the future. We always work hard and never give up. The learning and research processes at UGM continue to run together and explore state-of-the-art innovations. To create a convenient, environmentally friendly, and pollution-free green campus, UGM has built a wisdom park that substantiates the teaching and learning process. The park per se is open to the public. UGM has also developed drinking water facilities throughout the campus, provided by Drinking Water Supporting Unit. This system is harnessed by the entire academic community of UGM. As the manifestation of its teaching endeavor, UGM operates various field laboratories for educational and research purposes. Furthermore, UGM has established and run a science technopark as an embodiment of its capability of inventing and commercializing a myriad of innovative health products, ranging from medical devices, herbal medicines, and healthy food.
There are now more than 118,000 cases in 114 countries, and 4,291 people have lost their lives.
2020 has been a tough year for anyone, and it is no exception for UGM. Campus activities are abruptly halted, student activities on campus are annulled, and everyone is forced to work, worship, and study from home. Lecturers have no choice but to learn how to teach effectively using the Information and Communication Technology or ICT. Learning and research on technology are strongly supported at UGM, and in fact, we have conducted various cutting-edge technological research. Community Service or KKN is one of the most important courses to complete the study program, which will be undergone by University of Gadjah Mada or UGM in 2020 while adapting online courses system. Supported by the Ministry of Education, UGM will continue its service within the society across Indonesia during the pandemic issue. Lecturers and researchers at UGM continue carrying out basic and applied research in many realms. For instance, studies in the health sector to cope with COVID-19 are being encouraged and accelerated as a form of UGM's commitment to mitigating disaster using scientific methods. One of the most innovative inventions regarding to mitigating the COVID-19 is Genos, a screening tool or early detector of patients exposed to COVID-19, which is able to identify the symptom through exhale within 80 seconds. Genos is highly reliable. It has a number of advantages as a COVID-19 fast detection tool. Its sensor is able to detect more than thousands of patients in the long term, while providing relatively fast results, non-invasive and affordable. In addition, its usage also applies non-rebreathing mask and disposable HEPA filter. Those challenges somehow direct or even force us to innovate and modify approaches. Success is a journey which necessitates smart work through learning, studying, and sacrificing. Despite all that, most of all, we love what we do.
The Faculty of Veterinary Medicine of Universitas Gadjah Mada was established in 1946. It was started from the Bogor Veterinary Medicine College. In 1947, a parallel class was opened in Klaten. Faculty of Veterinary Medicine has 4 programs: Veterinary Medicine Study Program, Veterinary Profession Education Study Program, Master of Veterinary Science Study Program, and Doctor of Veterinary Science Study Program. Various tasks and expertises of veterinarian are learned in 12 departments. Each department is equipped with advanced laboratories and modern research tools. The 12 departments are Department of Anatomy, Department of Biochemistry, Department of Pharmacology, Department of Physiology, Department of Surgery and Radiology, Department of Internal Medicine, Department of Veterinary Public Health, Department of Microbiology, Department of Parasitology, Department of Pathology, Department of Clinical Pathology, Department of Reproduction, Obstetrics and Gynecology. Besides an advanced laboratories inside all of the departments, this faculty is also supported with animal health education and training unit, used by university students and veterinarians for the sustainable learning activities. This unit is called UP2KH, which manage several livestock such as cows, sheep, goats, and horse. Various achievements have been obtained by the Solari Society, such as the Regan Tester Kit for chicken carcass. FVM lecturers discovered this tester kit. It has been patented under the name Durante and has been widely used by the community. Faculty of Veterinary Medicine lecturers regularly write and publish books related to the veterinary medicine world that can be used for readers in the animal husbandry, biology, and statistics. The university students has also obtained several achievements in the research, arts, and sports field, and also national and international organizations. Some collaboration in the national level has been made. FVM UGM is the member of Indonesia Veterinary School Association. An MOU has also been signed between the government and private sectors for the purpose of research and community service. As for the international level, FVM UGM has also collaborations with foreign universities from Australia, Thailand, and Japan for research and education and other abroad universities. Furthermore, FVM UGM accepts international students from abroad, such as from Malaysia. The commitment to develop science and community service is based on the veterinarian work principles, manusia.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, dear professors, ladies and gentlemen, and welcome to the third International Symposium of Global Physiology, Challenges for Physiologists in the Post-COVID-19 Pandemic Era through the Development of Innovative Science and Technology, brought to you by the Indonesian Physiological Association. My name is Karisma Dewi, and it's always been an honor for me to be your MC here today. And we are delighted to welcome the 200 participants and more. So, before we start, we'd like to remind you some guidelines for you to follow in this plenary session. Ladies and gentlemen, you are advised to rename the display name into the following format, as you can see on the chat box as well. Full name _ presenter if you are going to present at the parallel sessions or participants. Kindly turn off your microphone and please turn on your video and adjust the speaker volume. All presenters and participants must use a virtual background that has been prepared by the committee by clicking the link that provided on the chat box as well. Later on, please fill in the attendance form that will be available in the chat box. Ladies and gentlemen, moving on to the next agenda. The first one is the national anthem of Indonesia, Indonesia Raya. The second one is Hymn of Gajah Mada, as today's event is taking place at Universitas Gajah Mada. And last but not least, the Hymn of Indonesia Physiological Association. So ladies and gentlemen, please welcome. Professors, ladies and gentlemen, there are more than 200 participants of today's event, and we are delighted to have you on this event. Therefore, to welcome you on the third International Symposium of Global Physiology, we are going to have several welcome statements. The first one will be coming from the chairman or the chairwoman of the symposium. So, ladies and gentlemen, please welcome Dr. Hewan Claudemona Airin, Magister Pertanian, Ahli Ilmu Faal Hewan.
Good morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. Welcome to the International Symposium on Global Physiology 2022. International Symposium on Global Physiology is a regular biennial meeting. However, because of the ongoing COVID-19 pandemic, the third International Symposium on Global Physiology is organized this year after 2 years postponed. The symposium this year will raise the theme of challenges for physiologists in the post-COVID-19 with development of innovation in science and technology. I am very excited to meet colleagues and researchers from various areas, although from virtual meetings. We have 6 great keynote speakers in this symposium coming from 6 countries: India, Malaysia, Japan, America, Taiwan, and Indonesia. We do hope that it could provide us with a good space for sharing information, knowledge, and research findings. Organizing committee members have worked extremely hard last year, especially in the pandemic era, to organize this symposium. For that, on behalf of the committee, I would like to thank all of the committee members of Indonesian Physiology Society, sponsor, and partner who have supported the activity. Thank you very much and enjoy your symposium. Wassalamualaikum warahmatullahi wabarakatuh.
Dr. Mona, thank you very much for your welcoming speech. Up next, ladies and gentlemen, we are going to have the welcoming speech from the head of Indonesian Physiological Society Chapter Yogyakarta. So, ladies and gentlemen, please welcome Dr. Slamet Widianto, Sarjana Sains, Magister Sains.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Honorable Professor, Lecturer, Researcher, ladies and gentlemen. On behalf of Indonesian Physiology Association Chapter Yogyakarta, I would like to express my sincere gratitude and welcome you to the third International Symposium of Global Physiology. The theme of the symposium is challenges for the physiologists post-COVID-19 pandemic through the development of innovative science and technology. Due to COVID-19 pandemic issues, people give a huge concern on how to increase their health quality. They consume health-enhancing agents such as supplement, nature product, or traditional medicine. Traditional medicine play an essential role in healthcare, especially in primary healthcare. According to the WHO, there are estimated to be used by about 60% of the human world population. In this symposium, we hope to able to provide a lot of knowledge and experience about the post-COVID-19 pandemic from the research from researchers. In this symposium, we continue the tradition of uniting the spirit of scientists, providing a communication between scientists is the best means of strengthening physiology knowledge and promoting the ideal innovation development. Thank you for your patience and participation. I also thank for Advisory Board, Organizing Committee, International Scientific Committee, institution, company, and volunteer for working hard to make this symposium a success and come to reality. I wish you a very productive symposium and have a fruitful exchange idea of physiology and provide valuable research to support the development of knowledge and innovation to overcome the COVID-19 pandemic. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Moving on from Yogyakarta, ladies and gentlemen, we are going to Bogor. So to welcome you, ladies and gentlemen, please welcome the head of Indonesian Physiological Society Bogor, Dr. Hewan Hera Maheswari, Master of Science.
Dear honorable speakers, guests, sponsors, and participants, Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Thanks to Allah, the Almighty God, who give us opportunity to attend a very important event, the third International Symposium of Global Physiology that will be virtually held on January 19 to 20, 2022. I'm Hera Maheswari, the Chair of Indonesian Physiological Society Bogor Chapter. First and foremost, it is a great pleasure for me to welcome you all, His Excellency the Dean of Faculty of Veterinary Medicine Gadjah Mada University, Professor Teguh Budi Pitoyo, Her Excellency the President of Indonesian Physiological Society, Dr. Ermita Ibrahim Ilyas, the Honorable the Chair of Indonesian Physiological Society Yogyakarta Chapter, Dr. Slamet Widianto, and the Chair of Indonesian Physiological Society Aceh, Dr. Golib. In particular, I'd like to extend my gratitude to our honorable invited speakers. I know one of them very well for many years, Prof. Iswal Pahar from RMIT Malaysia. Hope he is in now. How are you doing, Prof? I hope you are doing well. I would also like to extend a warm regards to all distinguished presenters, participants, and also all the technical team who make this event possible. Regarding the world is still in a pandemic due to the coronavirus disease 2019 or COVID-19 outbreak that has been spread globally until now and has its impact to the limitation of social movement. This third International Symposium of Global Physiology is carried out in the form of virtual conference. Ladies and gentlemen, I think everyone agree if I say that this symposium is a very beneficial for all scientists, researchers, and students in the field of physiology to show and share knowledge, information, technique, as well as experiences of their latest research, and also provide a good learning atmosphere to study and collaborate between scientific community in Indonesia and all over the world, in particular amongst the participants, guest speakers, and all of us here. Ladies and gentlemen, the new globalization and development in the 21st century have a tremendous effect on every aspect of our life. There are rapid change and development in many aspects, including science, technology, and communication that force every field of science to adapt and also give an opportunity to all fields of science, including physiology, to give more contribution to make a better life for every people. The improvement of information and technology system make distance not become a barrier anymore for the development of science and education. In addition, the era of big data enables every science analyze large amount of data such as genomic, proteomic, metabolomic, transcriptomic, rapidly or so-called omic sciences. This technology helps scientists to find a more accurate treatment for many diseases and predict the spread of infectious diseases that enable scientists to unlock some mystery of life. The 21st century is era of collaboration, not competition. We face different challenges than the previous era, such as global warming, climate change, non-communicable diseases, COVID-19 pandemic, or maybe some other infectious diseases that will emerge or re-emerge in the future. It cannot be denied that we cannot solve all the problems alone. We should unite and collaborate. Since the last few decades, there is a concept called One Health. In the concept of One Health, all aspects in human medicine, animal medicine, or veterinarians, and environmental work together because the human, animal, and environment health are interconnected. As I mentioned before, that the world are facing many problems such as the emerging of several infectious diseases, climate change, food security, food safety, etc. Those problems are not a single factor but multi-factor. All of us should collaborate to find the best and comprehensive solution to solve the problem. I believe this symposium will give us a positive impact, will increase our insight and knowledge regarding the theme of this symposium. Last but not least, again, thank you very much for your time for being here with us. I wish you to have a pleasant and fruitful symposium. Wassalamualaikum warahmatullahi wabarakatuh.
Dr. Hera, thank you very much. We are going to see you again later at the plenary session, yes? Ladies and gentlemen, since today's symposium is taking place at the Faculty of Veterinary Medicine, Universitas Gadjah Mada, the Dean of the Faculty of Vet Med, Universitas Gadjah Mada, would like to welcome you. So therefore, please welcome Professor Dr. Hewan Teguh Buditoyo, Doctor of Philosophy.
Waalaikumsalam warahmatullahi wabarakatuh. Dear colleagues, professor, lecturer, researcher, ladies and gentlemen. On behalf of the Veterinary Medicine Faculty, Universitas Gadjah Mada, Yogyakarta, I would like to express my sincere gratitude and welcome you to the third International Symposium on Global Physiology 2022. Moreover, I honorably welcome to all of our invited speakers. Professor Mei Ling Se, National Cheng Kung University, Taiwan. Professor Toshiya Muranaka, Osaka University, Japan. Professor Iswar S. Parhar, Monash University, Selangor, Malaysia. Dr. Rachel Sabine Kelly, Brigham and Women's Hospital, Boston, USA. Vijay Pal Singh, PhD, GSIR Institute of Genomic and Integrative Biology, New Delhi, India, and Dr. Diki M. Rizal, Department of Physiology, Medicine Faculty, Universitas Gadjah Mada, Yogyakarta. It is my hope that the third International Symposium on Global Physiology 2022, with the theme of Challenge for Physiologists Post-COVID-19 Pandemic with the Development Innovative Science and Technology, would be able to achieve its objective to provide a platform for academia and researchers to share their knowledge and idea in physiology and related fields, to build a professional networking between physiologists worldwide, as well as to strengthen the collaboration between researcher, government, industry, and non-government organization and NGO. I wish you a very productive symposium with exciting and encouraging discussion and exchange of knowledge so that together we can anticipate challenges for physiologists post-pandemic of COVID-19 by the innovative science and technology. The last but not least, my deepest gratitude goes to the advisory boards, organizing committee, symposium committee, institution, companies, and persons who have directly and indirectly supported the success of this international symposium on global physiology in Universitas Gadjah Mada, Yogyakarta. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Prof. Teguh. And to begin the symposium officially, ladies and gentlemen, joining us from Jakarta. Here we go, the head of Indonesia Physiological Society, Dr. Ermita Ibrahim Ilyas, ahli ilmu faal olahraga.
Thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Dear Honorable Professor Dr. Hewan Teguh Budi Pitojo, the Dean of Faculty of Veterinary Medicine Gadjah Mada University, Dr. Slamet Widianto, President of IPS Yogyakarta, Dr. Hera Maweswari, President of IPS Bogor, Professor, College, and ladies and gentlemen. Good morning. Peace for all of us. First of all, let us praise Allah that we can gather here together in the virtual symposium organized by IPS Yogyakarta to attend the opening of the third International Symposium of Global Physiology, January 19 and 20, 2022, with the theme Challenges for Physiologists in the Post-COVID-19 Pandemic Era through the Development of Innovative Science and Technology. In this occasion, I would like to thank and give a big appreciation to our dear distinguished guest speakers, Pinjay Pal Singh, PhD, from CSIR Institute of Genomic and Integrative Biology, New Delhi, India, Professor Toshiya Muranaka from Department of Biotechnology, Graduate School of Engineering, Osaka University, Japan, Dr. Diki Muhammad Rizal, Spesialis Andrologi Master Kesehatan from Department of Physiology, Faculty of Medicine, Public Health and Nursing, Universitas Gadjah Mada. Rahel Sabin Skelly, PhD, from Department of Medicine, Brigham and Women's Hospital. Professor Iswar S. Farhar from Brain Research and Neuroscience, Jeffrey Chia School of Medicine and Health Sciences, Monash University, Selangor, Malaysia. Professor Meiling Tsai, MD, PhD, from Department of Physiology, Medical College National Cheng Kung University, Taiwan. Dear all participants, we know that we have gone through the COVID-19 pandemic for 2 years already. So this condition influences the performance of the physiologists around the world, especially in the research area. We know that there are still many challenges in the research area in physiology, and we really hope this pandemic will end soon. So I hope the speakers in this virtual symposium will show us many spectrums of research in the future after the COVID-19, the crisis or ends, to encourage our participants in this morning to prepare the proposal after this symposium, hopefully. Ladies and gentlemen, I would like to thank and also give a big appreciation to the organizing committee for their hard work in preparing the symposium, especially in this condition. Working hard to prepare a virtual symposium for us to maintain the scientific environment, to remind all physiologists in this country to have a big hope for the future of research in physiology area. Thank you to all dear colleagues. And for all participants, I hope this symposium can give you useful and valuable information and knowledge to bring home. But don't forget to stay and keep healthy. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Waalaikumsalam warahmatullahi wabarakatuh. Dr. Ermita, I would like to invite you to officially open the virtual symposium.
A great pleasure to welcome you to the third International Symposium of Global Physiology 2020 with the theme Challenges for Physiologists in the Post-COVID Pandemic Era through the Development of Innovative Science and Technology, held here in Yogyakarta. And a great honor to open the third International Symposium of Global Physiology 2020. Thank you.
Thank you very much. Since this is a virtual symposium, it's a little bit hard to hear the clap, the applause sounds from the participants. So you are invited to give the reactions by clicking the reaction icon on your screen below. Ladies and gentlemen, as already mentioned before, the third ISGP topics would be about the challenges for physiologists in the post-COVID-19 pandemic era. And there are 3 invited speakers for today's plenary session. And Dr. Hewan Hera Maheswari, Master of Science, would lead the discussion. A brief information about her, ladies and gentlemen, as you can see on your screen. She is a lecturer at the Department of Anatomy, Physiology, and Pharmacology, IPB University. She got her veterinarian degree at the IPB University as well, and then Master of Science in 2000 from University of Queensland, Australia, and Doctor of Reproductive Biology in 2006 in IPB University. Her research publications, of course, are more than that, but one of those are determining image opacity in broiler respiratory radiographic. As you can see, that has been published in Journal of the Indonesian Veterinary Research. So joining us from Bogor, West Java, ladies and gentlemen, please welcome Dr. Hewan Hera Maheswari, Masters of Science.
Very much, Charisma, my dear Charisma, our Master of Ceremony. Yeah. Thank you. Introduce myself. Hello, everyone. Assalamualaikum warahmatullahi wabarakatuh. Hope you are all in good health, good spirits, and of course in happiness always. Okay, it's a great pleasure for me to lead this first agenda of our symposium, the plenary session, day 1. In this plenary session, we will have 3 invited speakers, Vijay Palsingh, PhD, from New Delhi, India, and Professor Toshia Muranaka from Osaka University, Japan, and Dr. Diki Muhammad Rizal, Specialist Andrology EFM, Master Kesehatan, from University of Gajah Mada, Indonesia. I think I would like to remind you before we start the session for you all to switch your mode of a laptop and gadget into the silent mode to avoid any disturbances during the presentation session. Before we start, I would like to introduce you to Dr. Vijay Pal Singh. As you can see on the screen, Dr. Vijay Pal Singh is Associate Professor, ACSIR and STO at CSIR Institute of Genomics and Integrative Biology in New Delhi, India. His formal education graduate at Veterinary Medicine, Gujarat Agricultural University, and he's got his postgrad on dairy husbandry and milk processing, PTC Plus in Netherlands, and he's got his PhD in biotechnology, Savitribai Phule Pune University. Hope I spelling in the right way. India, doctoral biotechnology. And his research publication, I think this is the latest one, is about downregulation of mRNAs during delayed wound healing or in diabetes role of DICER. And you can find this publication on molecular medicine. So now I would like to invite Dr. Vijay Pal Singh.
Hello everyone, good morning. Thank you very much for the nice introduction. Can you hear me?
Yes, of course.
Okay, let me start my sharing my screen. Can you see my screen?
Yes.
Okay. Good morning, friend. And this is Dr. Vijay Pal Singh from India. And as you can see, it is early morning here in India. And there is a lot of cold here. It is 7 a.m. and it's very, very cold in Delhi. But today what I'm going to talk, and before I start my talk, I would like to thank the organizer for the opportunity, especially Professor Astuti, because I think we met long back in Bogor University, and since thereafter we are colleagues together, and because of that reason she has invited me to this virtual conference. And as in the inauguration have been pointed out by the Professor Armita about physiology, I am going to talk about more of the animal experiment that we plan for physiology or for some other experiment, including COVID. As you might have seen that earlier in the time, the vaccine used to take 100 years. And now one vaccine, which is against the COVID, at least in India, we have 2 vaccines which are made in India, have come out within a very short span time of only 1 year, or around 1 year, or less than 1 year, because we could use animal. Now, before I start, I am very interactive person, and usually what I do a lot of interactive talk in the class, but in the virtual mode, it will not be possible. Hence, I'm going to talk only for the purpose of means not interactive, but kind of a one way. So, and before I start, I would like to tell you the brain is a wonderful organ created by God, and it starts working as soon as you wake up. You wake up and your brain is starting to work. But the issue is that it stops once you enter into the webinar. So all of us, we are having a lot of webinars and online conferences, and you come to the webinar and you think, oh, now and then you stop your video and then you go for dreaming. So please don't do that. Here we are going to have a very wonderful discussion on animal experiment design for physiology. So with that note, let's start. As you might have seen that there are a lot of animal experiment failure. From this cartoon, you can see that experiment failure is huge, and because of this reason, the people are started to blame that yes, animal experiments are not successful, but it is not the animal experiment will fail, but it is a human experiment which also fail. Like phase 1, phase 2, phase 3 are the trial, preclinical trial are done on animal, but phase 1, phase 2, phase 3 are done on human. So still there is a failure. You can see from the cartoon that there is a failure even to phase 1, phase 2, and phase 3. So it is not the human experiment, animal experiment failed, but also the human experiment also fails. And preclinical research which is done on animal is always saving us from the death because of the toxicity. So it is because of this reason that we don't die, the animal are being used and they save us from the toxicity, profile and toxicity death. Now coming to the statistics and ethics, why most published research findings are false. This is the recent paper in PLOS Medicine which says about the published by John P. A. Ioannidis, that why our published research findings are false means we are always fixated towards p-value. P-value is less than 0.05. Now it is significant, hence it is useful. No, that's not true. Unless we have the power of our experiment, power of animal experiment, this is not going to be very useful. So most published research findings are false for the reason that they are not being taken at animal experiment which is with enough power, that we are going to discuss in detail. Now, what is an animal experiment? Animal experiment basically has three components. First, it is performed on living vertebrate. First of all, two important things, living and vertebrate. Living means those animals which are living being, not dead. If something you are doing on dead animal, or if you are taking the tissue from the dead animal, it means that experiment is not animal experiment. And also performed on living vertebrate. In that case, if you are doing something on Drosophila, this is not considered an animal experiment. Then it is causing some distress or suffering to animal, means that it is causing some pain and suffering to animal. If there is no pain and suffering, simply you are taking blood for animal health monitoring, it is not an animal experiment. And also it is done for certain purpose. And purpose cannot be your degree, like your master degree or PhD degree, but purpose have to be very, very specific, that you are going to solve some societal problem. Hence, three important things: perform on living vertebrate, cause some distress or suffering, and is also done for certain purpose. With this, why are we caring so much for animal experiment? 300 times more consumption of fish more each year than the usual catch in our research. Household cats approximately 5 million animal work than the animal used into animal used in our research. And also you see the consumption of chicken in a year is more than total number of animal used in medical research since past 2 centuries. So we are consuming fish, we are consuming chicken, and we are also, our household cat is also killing so many rats. Why are we worrying about animal which is in lab?
Halo Mbak, halo.
Yes. Hello.
Iya, aku ketendang iki. Aku ketendang iki. Aku ketendang saka laptopku.
Hello, can you hear me? I see some background noise. Hello.
Hello.
Yeah.
Hello.
Yes, sir.
Sorry, your slide is still not move. It's still number one.
Excuse me.
Slide at the same.
Your slide is not mixing in.
Okay, let me do it again. Let me share again. Is it moving now?
No.
No, okay.
Yeah.
Wait.
Doctor. Would you like to click the slideshow icon on your presentation? Slide, yes. Yes, we can see it. Now, would you like to click the slideshow icon? It's moving from the first slide, yes, but it's still not in a full screen mode. Oh, okay. Yes, okay. Yeah, thank you. I'm sorry for the interruption.
To move it,
Now I'm trying to move.
but it's still not moving.
What to do? Can I send these slides to you?
Oh yes, of course, to the committee. I think like maybe Charisma, can you help us?
I have sent the slide. I think, and by that time, let me try it again.
Yeah, it's okay.
Yeah.
Time.
I am sure this is. It is coming. Whiteboard, iPad, screen, power slide. Can you see now it is moving?
Yes, good.
Okay. Thank you.
Yes, okay. Yes.
So I think we were here. What is an animal experiment? So animal experiment has basically three components. As I told you, for form on living vertebrate and cause some distress or suffering to animal, and also is done for certain purpose. These are the three very important component of animal experiment for done for some physiology or for some other experiment. And if these three components are not there, we cannot classify this as an animal experiment. Why we care? As I discussed with you, that 300 more time consumption of fish, chicken and everything. Why are we caring so much about animal experiment? Because animal experiments are not done for animals most of the time. It is done for human. If we are doing research on COVID vaccine, it is not for humans, animals, sorry, it is for us. And it is because of this that they are important. And if we are not doing this experiment properly, that these are the incidents which will happen, like TGN1412 monoclonal antibody case, where the B-cell lymphocytic leukemia and rheumatoid arthritis, they were tested on mice, rats, and cynomegalo macaque, and the dose was 500 times smaller. And in the were given into 6 per volunteer induced life-threatening condition. So remember, it was already tested on mice, rat, and monkeys, macaque monkeys. Dose was 500 times less. When it was given into human, 6 volunteers, it caused life-threatening condition, and many were died. So if you just Google TGN1412 case, you will find such kind of a thing. And this is the problem, that if we are not doing animal experiment properly, these are the consequence, and this consequence is happening in human. And because the CD28 and T cells were organ differentiation and activation was a bit different in mice and rat, as mice and rat present less memory T cell than the humans, and also macaque CD4 T cells lose expression of CD28 when differentiation, but no human CD4 T cell. So, because as I discussed, these are the failures which will happen. If there is a failure in corona vaccine, which is done on animal like ferrets, as well as on this animal mouse, which are having ACH2 knockout receptor, then the vaccine failure will happen into the human. And that is the basically biggest problem. Hence, as we are doing research for humans, we are very careful in animal experiment. And we should be careful. Otherwise, if we are not careful, animal will have some consequence. But ultimately, this bad effect will come to humans, it will come to us, and hence it is important. And can we generalize this study? What are the basically issue in our animal experiment? Now I am coming to what are the problem area, why there are a lot of failure, as we discussed that there is a lot of failure in animal experiment, and because of that we will discuss some of this issue. The issue is that can we generalize from our study that usually we have to use both male and female, but most of the time you will find the scientists are only either using male, and still male is the dominant in animal study, or otherwise female. But then, which I will discuss in detail, the male, if we are doing the generalization of animal experiment. Generalization means a vaccine which works in animal, which also will also be working in humans. A vaccine which is working in mice will also work in other species. So that we call as generalization. Inbred and outbred strain. Mostly we are using inbred strain, and outbred strain are out of the question. Basically, we are using outbred strain basically for toxicological research. Yes, it is important. So inbred strain are those strain where we are continuously breeding the mouse for 20 generation into brother and sister. So they totally become genetically similar, same genetic. So inbred and outbred have to be used very, very consciously. We are all the time mostly using rat and mice, but not at all human. And randomization and blinding is very, very rare in scientific experiment that I am also going to discuss in detail. Randomization means that you are whatever the difference, like male, age, sex, weight, everything you are distributing among all. And blinding is this: when you are doing animal experiment, you have to blind. Blind means that you don't know what you are doing, and you also don't know the outcome. If we are knowing that I am doing anti-diabetic drug testing on animal, then what will happen? You will always find out some result out of this, which is not a very, very good idea. And experimental unit, it must be possible. Experimental unit is that it must be possible to assign any of the two experiment unit to different strain. Here the issue is this, I hope you can see me, that if you are having a case, and in one case you are having 6 mice, and these 6 mice are placed near to the one light, but these all 6 mice are of control, then your n is equal to 1. Because they are not independent. All 6 from one group are exposed to one light source, and they are exposed to sometimes high light or lower light. So it is important that you distribute your experimental unit to all the possible group, one from the control, one from the treatment, one from the high dose, one from the mid dose, one from the low dose. So if there is some variation which is there, it is getting distributed among all the group. Now coming to observation. Observation is very, very critically important. If you see, I will give you two examples. Vincristine, initially it was originally meant to be anti-diabetic. It was not meant to be anti-cancerous, but it is very, very popularly used anti-cancerous drug for our veterinary sector as well as for the human sector. Also, tamoxifen, which was initially was for birth control, then the antagonist to the tamoxifen. It is also now being used as anti-cancerous drug. So unless you are observing with the open eye, not only the things which you are doing, because most of the discovery in the drug discovery case have come up because of the observation. So vincristine and tamoxifen is the example. So don't you only see that my drug is anti-diabetic, but if you see something outstanding, some research which is out, means observation which are totally different, you have to also observe that. What are the activities that animal has started to do? Mostly example like paracetamol, which is there, and also this example of a very popular other drug is there only because people were observing. Penicillin is another example. So unless if you are observing animal very closely, you will not see the difference, and observation have to be very, very important. Changing what is normal require us to change the lens we are using. This is the biggest problem, my dear friend, that in physiology or in other experiment or animal experiment that we plan, we always think from our point of view. We have to think from animal point of view. What is that animal point of view? We like to be in a very good environment, air-conditioned 5-star hotel. But for mouse, the 5-star hotel is different. They like to be in their burrows. They like to be in their own environment. They have their own smell. They have their own good smell. But we consider that good smell of mouse is bad. No, that's not true. So it is very important that we look from the point of view of animal, what animal want. In case, suppose if you are saying you are doing experiment on pig, what pig want? Pig want is that they like to always do the snouting. Snouting means they like the mud, and in the mud they will do snouting. So if you are doing experiment, recently if you might have seen that heart is being transplanted from the pig to the human successfully. So if you are having the pig as your lab animal, you have to consider what they like. Same is also true for poultry. What they like roosting. So mouse, what they like, burrowing. And they also like the socializing. They also like to travel. They also like to have always the company. So it is important when you are planning the experiment that you have to start looking from the animal point of view. We have to do a standardization. So this is the actually practical exercise that I used to do when I'm not doing physical exercise. Professor Astuti knows, then it is very funny things that we do together. So why do we standardize? Standardize means whatever the experiment you are doing in Yogyakarta or into the Bogor University, and I'm doing in Delhi, should be reproducible, and others should be in the position to achieve the reproducibility. How can you do that? This is the runway, and every plane which comes will only land into this runway, and reproducibility is very, very high. So why it is so high and why it is so precise? Even though there are, if a plane is flying onto this in the sky, they also have a lot of variation. They have weather which is unpredictable, and they have the issues which are totally unaware of, but still they always try to pop up, and they always try to be successful. How can they do this? Because they always follow the checklist of 10 to 15 checklist, even on a very short routine flight. Even if I'm 50-year-old and if I'm flying a plane, I have to do a checklist. I've done this, I've done this, I've done this, I've done this, and then only I can fly. This is something which is we are lacking. We don't do a checklist and we think we know everything. So there are two checklists. One is the arrive guideline and one is the prepare guideline. Prepare guidelines are those checklists that we have to start before we start our animal experiment. Prepare guideline. And arrive guidelines are those guidelines which we are doing when we have finished our experiment. So before and after. Why are we doing this checklist? Because we are flying, as we are flying like an aeroplane. So we have to have our experiment and we have to have our checklist, whether we have seen health, whether we have seen diet, whether we have seen the body weight, whether we have seen the environmental condition like temperature, humidity, and all this issue. So these are the things which we have to see as a checklist, which is very, very important. But this reduces our risk of forgetting to carry out the vital actions. Ensure checks are carried out in correct sequence. Suppose I give one example. If you have seen the animal and if you have only done the kind of a health checkup when experiment is over, mycoplasma infection was there and you did not check and you have completed the experiment, you can't do anything because it has to be done before you start an animal experiment. So you should know the correct sequence and you don't forget. If you have a checklist, you encourage the cooperation and cross-checking between cruise member and make sure that everything is on the same page. So this is the way that we have a checklist for animal experiment that we call as prepare guideline. And after experiment, we call them arrive guideline. That is for reporting animal experiment. And it is because of this checklist is very, very important. But don't do checklist when you have already released into the Bogor or into the Indonesia. You have to do it before you fly from Delhi. If you are flying from Delhi to Bogor or to the Indonesia, checking has to be done before, not after arrival. So this is the case in our animal experiment, that if we finish our experiment and sometime we forget what we supposed to do and we have not done, and hence our results are all not very useful. Hence, because of this reason, we have to have standardization and we also have to check a checklist before we start our animal experiment. And I told you, sorry, these are the checklist, prepare and arrive guideline, which we have to see. Now coming to the value of animal. So animal have two kind of value, extrinsic value and intrinsic value. Sorry, extrinsic value are those value which are available to us. Like if you are having buffalo, you get a milk. If you have a buffalo or a cow, you get also the meat. From the goat, you get a meat. So that is the extrinsic value of that animal to us. So in our lab, animal, the extrinsic value is what? Extrinsic value is the data. So data is the extrinsic value of laboratory animal. And what is intrinsic value? Intrinsic value is inherent worth, my importance, my values, and that exclude utility. And they are basically connected, extrinsic value and intrinsic value, and they are connecting in the way that if we don't provide the intrinsic value which is important, like for the birds it is roosting, for the pig it is snouting, for the mouse it is burrowing. If we don't provide that, then the extrinsic value will not be useful. Extrinsic value is data. So suppose if you are doing experiment for corona or COVID vaccine and your experiments animal are stressful, then your data which will come will not be useful. So it is that important that extrinsic value and intrinsic value have to be taken care. Then validity, internal validity and external validity. Internal validity is the one where you can establish cause and effect. Cause is temperature, means you have a fever, and effect will be bringing down the temperature from the paracetamol. So you can establish what is the cause and what is the effect, what is dependent variable and what is independent variable. Dependent variable is temperature, independent variable is paracetamol. So if you are using paracetamol, temperature should come down. So internal validity means whatever you do, it has to be reproducible, it has to be we can establish cause and effect. External validity is those validity where our experiment or our experimental animal can be used for other purpose. Means if you are doing experiment on mice, they are not for mouse, so they should basically for humans. So if you can do experiment, it should be useful for other species. Sometimes we are doing for experiment for animal also. So in that case, experiment have to be useful for our buffalo or for our animal as well as for some other species. So internal validity, you can establish cause and effect. External validity is the one where you can say that my experiment is going to be useful for human as well, other species. So this is called a generalization. So internal validity, external validity. It is like same, if you are having a valid license, then you can only drive the bike. If you don't have a valid license, you cannot drive the bike. So that is the importance of this internal validity and external validity. As we have discussed, you should be able to establish the cause and effect or dependent and independent variable, and there should be no confounding factor. Like if you are saying, I am discovering a drug for cancer, you should be able to establish that effect which is coming to reduce the cancer or cancer size. It is because of my drug, it is because of my intervention and not by any other factor. Any other factor means because of the diet, because of the temperature, because of the environment, because of the animal genetics. Sometimes animals are robust, they themselves get cured. Suppose if you are inducing diabetes by estrogen injection, sometime animals, and most of the time the estrogen and beta cells try to recover, and they recover by themselves. It is not because of your drug. So that is the confounding factor. The recovery of diabetes by confounding factor is that they recover by themselves because the destruction which have happened to the beta cell is by only your intervention is temporary. And suppose if you are doing treatment for temperature, paracetamol kind of a drug, and you have already a condition, so animals started to have less temperature because of the outside environment. Sometimes you have estrogen into your diet, which is also affecting and also causing a lot of trouble. So if you establish cause and effect, no confounding factor, it means perfect, your experiment is internally valid. Sure. So importance of experiment design is good experiment design enable the investigator to control for threat to internal and external validity. Threat to internal validity compromise on the confidence in saying that dependent variable were caused by independent variable and not by some other variable. So when you say cause and effect, so you can definitely say that this effect is because of my drug, not by any other factor. Then your internal validity is okay. Threat to external validity compromise our confidence in stating whether study results are applicable to other group or to generalize. Means whether our experiment can be generalized to other species and to other humans also. These are the issues related to internal validity. So how do we address them? Selection bias. Sometimes we select some animal and then we use them, like we use only 8 to 12 weeks, or we use 10 to 15 or 20 gram of mouse. So that can be avoided by randomization. Suppose if you have the range of the weight from 20 to 30 grams of animal you have, then each group should have 20 gram of mice, each group should have 30 gram of mice, each group should have 25 gram of mouse. So that weight range which is there has been randomized is going to everyone. And performance bias is allocation concept. You should not know, you should be blinded that what is the performance. You just note down, but don't see performance by group. Otherwise, you will try to find out some effect which is there. Detection bias, you should always be blinded about this. And attrition, if some animal are dying, then you have to justify, but don't replace this. External versus construct validity. External validity refer to the ability to generalize the finding to different measure settings and time. Construct validity etiology refer to the adequate representation of theoretical construct in the disease modeling, and this may be threatened where only specific characteristic of complex disease are modeled. So here there are two complex disease like asthma. It is very complex. So you cannot You cannot have a complete model for asthma. In that, you are trying to mimic some features like airway hyperresponsiveness. We get like when we are breathing like this in asthma, our respiration is high. So we try to only mimic that. Suppose if you have a corona, we cannot have the mouse or a Vista rat for doing the corona testing. We have to have a specific mouse where that receptor, ACH2 receptors are there. So these are the knock-in and knockout receptors which are there in the mouse and which is available from the jacks. And also some of the ferrets which are used for animal experiment. And these, because of this reason, the constructability means some agent, some physiology same into animal as well as same into our mouse model. Phase validity is a phenomenological or pathological similarity, means whatever the pathophysiology which happen in humans should also happen into the animal, or phenomena like in diabetes, the blood glucose go high. Such kind of a phenomena is called as pathological similarity. Predictability or response to medication. If you are giving paracetamol to the cow, the temperature will come down. If you give paracetamol to humans, temperature will come down. So this is called as response or predictability, which we can say. So these are basically the statistics. I'm not going to discuss much in detail. Statistics is the science of collecting, organizing, summarizing, analyzing, and making inference to the data. You people all know central tendency, like variability around the tendency, and that we can see. And also from the difference between the group, that is recession between the group, that is the inferential statistics. So these are the common type of data, qualitative, quantitative, and all this. But what I'm coming to you in detail, which is the biggest problem in our laboratory animal experiment, is that we always take how many number of animals? 6 number of animals. But is this 6 number of animal okay? No, it is not okay. So there are two ways. Either you have to do power calculation when you plan your experiment, that we are also going to discuss, and you have to have enough beta. Beta means power, that this effect which is there in the experiment is to be captured properly and also very, very predictable. You have to also describe the signal. Signal is the effect size or expected effect on central tendency, and that is the difference between your control and your experiment that you are planning. And noise is the intrinsic variability, the spread around the central tendency. More the variability, more the sample size. So we don't want variability. What the variability? Variability come from the temperature, room lighting, room feed, animal health, and also animal weight and some other issue. So basically 4 or 5 things are important for sample size. Alpha, we usually fix at the 5. Beta, which is 1 minus beta, which is the power to capture the difference which is there, which we are fixing at 80 to 90%. Signal is the effect that we want. That is we have to fix the effect, that what effect that we want from them, that has to be first fixed by us. Suppose if you have a cancer, my tumor size is this into the animal. Suppose this I'm just saying, just for the after the treatment, the size of the tumor, suppose this is 5 centimeter. After the treatment, that size will reduce to 1 centimeter. So your effect size from 5 centimeter to 1 centimeter. So what is your effect size? 4 centimeter. Suppose if you are doing research for temperature, high temperature, then you say, okay, in high temperature, my animal are having higher fever, like 105. After my treatment, that 105 temperature will come to 101. So effect size is 4. 105 to 101. So that is the effect size. You have to decide before. Suppose if it is corona, before experiment, animal are having such a load of virus that we can calculate through CT value. But after my treatment, the CT value or the load of the virus will come down to certain value. So this is the effect size. This you have to describe before your experiment. Noise is always there, and noise is the killer of animal experiment. What is noise? How noise? Noise is coming from the feed, noise is coming from the water, noise is coming from the health, noise is coming from the infection, noise is coming from the light, noise is coming from the food, noise is coming from the technician who is handling, noise is coming from the injection who is giving. So all these things have to be avoided. So remember, alpha, beta, signal, and noise. These are the things which we should know. Don't take 6 is to number of animal, which is good for all your experiment. Suppose you have a wonderful drug and you have a very good signal. Signal is what? Signal is effect size. What is effect size? Effect size is reducing temperature from 105 to 101, reducing the cancer from this size to this size. This is your signal. Signal is good, your drug is good, but noise is very, very less. Means you have exactly selected the right model. Suppose you have selected the right model for corona, your noise is very less, signal is very good. Then your effect size will be very good. But if signal is good, your vaccine is working, but you have chosen star rate for the testing of vaccine, then nothing will come out because there will be huge noise. Signal will be gone by the noise that you create. Effect size will be 0. This you remember. We have to reduce the noise and we have to increase the signal. How to reduce the noise? Noise can be reduced by taking very right animal, healthy animal, same size, same sac, the good temperature, light and humidity, same technician. So 100 of things that noise will be reduced and signal will be good. If you want your signal to amplify in your research, you have to select the right model and all these things which I am mentioning. Otherwise, your effect, which is their effect size, will be gone. It will not be useful. Sample size by power analysis. So this is basically, as I discussed, we have to decide the signal, and signal divided by noise is your standardized effect size. So what is noise? I am trying to tell you from the point of view of not a statistician, but a common man. Remember, my dear friend from Indonesia, statisticians don't know the biology and biologists don't know the statistics. That is the biggest problem that you have to connect, and this is very, very critically important. I'm not a statistician, I'm a doctor, but you have to understand the concept that what is signal, what is noise, what is significance level, and what is this power, so that we select proper sample size. If we are not talking proper animal sample size, then our vaccine or whatever the research we are doing will not be useful. Remember, again, I am pointing out this is very important. Signal, you have to decide that my signal will be 105 to 101, this size of cancer to this size, means such a load of virus to such a load of virus. This you have to decide before. Noise can be reduced by giving the experiment design with standardization. As I told you, standardization is also used by pilot. Checklist beforehand, health, feed, water, technician, these. Beforehand you start. So if you have a good checklist, noise will be reduced, signal will be high, signal high, noise low, some effect size will be very, very good, and you will have a successful animal experiment. Power is the 80 to 90%. As I told you before, why the animal experiments published research finding are false? Because we are not thinking of power, we are only thinking to significance level, 0.05. Our value is less than P 0.05, hence useful. No, we should have enough power. Power is 80 to 90%, and then you specify. I will also give a simple example. So you have a comparison of two anesthetic for dog under clinical condition. So these are the dogs that we are using, all British variety of dogs, Beagles, Old English, Griffon, Bull Terrier, and Irish Terrier. And we have decided their weight range of dog is from 3.8 kg to 42 kg. So huge variation. Remember, what is the weight range? 3.8 to 42.6. So their SD is very high. SD is standard deviation. Remember, SD is standard deviation, which is also we called as variation, or means noise, which we are discussing. So SD is 36. So signal is 20 mmHg. 20 mmHg. We want our blood pressure to come down by 20 mmHg. Here, the significance level, this alpha is 0.05, power is 90%, two-side t-test. Okay, so important is here that the SD is very high. What is SD? Noise. What is noise? Variation. And signal is, here it is 20. 20 is our effect size. What we want? What we want that by these two anesthetic under clinical condition, the blood pressure difference should be around 20 mmHg. So effect size is 20, SD variation is 36. So if you divide 20 by 36, signal by noise. What is signal? That is the thing that we want. Like in this one, what we want is our blood pressure to be low. So this what we want is 20, and what is effect size? 36. So our standardized effect size is signal divided by noise, 20 divided by 36. So in that case, what will be the effect? And effect will be around 0.56. So what is 0.56? If you see SN ratio, 0.56 is equal to around 6. Can you able to hear, please? I hope I am clear to you. Can somebody say yes?
Yes.
So if you see signal SN ratio 0.6, power 90%. How many animal you need in a group? 59 animal. With 80% power, how many animal? 45 animal. So nobody can give you the number which is 45. So because of this, we should not say our experiments are useful, and hence we cannot do this experiment. So with this number of group of animal, where the weight range is difference from 3.8 to 42, where the signal is okay, signal we have decided 20, but the variation is high. What is the variation? Noise. What is the noise? Noise is coming from all the source. In another experiment, we decided to use only Beagle's dog, no other dog, Beagle dog, uniform. So you see SD have gone down from 49 to now SD is 9. Effect size is same. Effect size is same, but the weight is also very I mean, should say less. Variation is less. The weight started from earlier it was 3 to 42. Now the weight is 17 to 23. SD is very less. So effect size is same, 20. 20 divided by 9, then comes 2.2. So in this 2.2, suppose if you see, you go to this 2.2, see this table. This is the Cohen effect size table. 2.2, how many animal you need in a group? 6. If you have 80% power, how many animal you need in a group? 4. So required sample size will be 6. This is the way we have to decide our experiment. Reduce noise, increase signal by selecting the right model, right number of animals statistically. This we call as sample size calculation. If we are not just taking care of anything but simply deciding 6, then our experiment is complete failure. Again, suppose if you have decided, if you are using 6 number of animal and you are using random dog, in that case you are means only you are right by 18%, and rest of the time like 82% you are wrong because your power is not enough. You are supposed to use 56 animal, means standard deviation was 36. You are supposed to use 68 animal. You are using 8 number of animal. Your power is 18% only, and your experiment is not going to be useful. Suppose if you are supposed to use 6 number of animal, you are using 8 number of animal, signal divided noise by 2.2, 98% of the time your experiment is correct. You are confident that 98% the finding that is there will be useful. I hope you can get it. I have already shared the slide with the organizer, Professor Astuti. You can circulate. If you have any confusion, please you give me a mail. I will also explain. I love to talk. I love to teach. If you are there with me by group, then I would have done with more interaction. But now this is only virtual. In virtual, you don't get excitement. You are sitting in one somewhere and you feel you are talking to yourself. Okay, so but this is the message I want you people should take from my talk. Sample size calculation have to be calculated. It has not to be okay, use only 6 number of animal model have to be selected, noise have to be reduced, signal have to be amplified. How signal amplification will happen? Right model selection. How noise reduction will happen? Standardization. How successful experiment will happen? Checklist. Remember, checklist means our pilot, those who are flying the plane. And this is very, very critically important that we should select these experiment and also these animal group properly so that our experimental use. This is the basically our issue with this what we call a sample size error. If we are using too few animal, means we supposed to use how many? 69. We have used how many? 8 or 6. It is total waste. Don't do such experiment or such experiment. If it is a too many animal, large number of animal, maybe it is a partial waste. So remember, I told you, statistician don't know the biology. Biology don't know the statistics. Important for you is that at least you know the nuts and bolts. You know the preliminary thing so that you can explain to the statistician. This is what I want. This is the parameter. This is the alpha. So if you talk in that way, you are on the same page. I'm not a statistician and I'm trying to tell you statistics is very, very important, and don't take just simple 6 number of animal per group. So we are making type 2 error, which is most common, as I discussed to you. Effect size is small. You are using 6 number of animal, out of that 1 will be dead, 1 will give very different result. Then what will you do? You try to normalize. One normalization means out of you supposed to use how many? Suppose you supposed to use 10. You have used only 6. 1 is dead. 1 is giving different result. You have left with 4 animal only. So effect will be very, very small. Variable is large or variation is large. What is variation? Standard deviation. What is standard deviation? Is noise. So noise, standard deviation and variation is one and same thing from the biological point of view. So variation is large, effect size is small. So then you start playing with the statistic. How can I bring my value? P value is less than 0.05. Test of significance. And also number of animal used is small. So this is the type 2 error which we commonly make. Our effect size is small, variation is large, number of animal used is also small. So in that context, our experiment will not be useful. And again, most people issues that we face in that we are not consistent what we say and what we do. So even if this is the research which has been done, people say something and people do something. Means they say, oh, we are taking care of animal very, very good and this and that. But when you go and see your experiment in animal facility, you will find something different. So your intention and action should match, because we are doing scientific research. Remember, my friend, if something is happen in Yogyakarta, it should also be reproducible in Delhi, in India also. So our intention and action should match. This is the one example I'm trying to find and give you, that experimental failure. 1026 drugs were tested for stroke in animal model. 374 were effective in animal. Only 1 drug was effective in humans. That was aspirin, which is already discovered drug. So repurposed drug. So this is a lot of experimental failure that you can know through this systematic view and meta-analysis. And in this, why they were a failure? Because in our experiment we have fixed thing. This is the stroke will happen, we will give the drug. But in real condition in humans, you never know. Now I have a stroke, but when will I reach to the hospital? The variation is there, time is there. So when you are planning experiment, you should also consider how things are happening in humans, should also replicate the same. If you don't know, go and sit with a doctor, human doctor, who are a stroke patient or a diabetes doctor or a cancer doctor or a vitiligo doctor or asthma doctor. Talk to them. What is the problem? How do you diagnose? Try to have same feature into the animal model and also see the systematic review and meta-analysis. And when in doubt, always support animal. Can we really call ourselves animal carer when we allow mechanization and complacency to perpetuate what is in rational context is cruelty? When you are doing animal experiment, don't be friend. You always discuss and positive criticism. Okay, sample size should be done, health monitoring should be done, animals should be selected properly. Wow, what is the etiology, whether in the COVID you have the ACE receptor there or not, whether this is the model which will good. Don't say I am friend, so I will not ask question. Critical discussion should be there. Otherwise, in what we call in the real context is the cruelty. Another issue is tradition and status quo. We are always doing it that way, or resistant to change, both individually and in the form of institutional inertia, all combined to challenge those who ask the appetitism. Sorry. So sometimes you say, oh, I am doing this last 20-30 years. No, this is not acceptable. It has to be discussed all the time, and because of this reason, we have ethics committee. An ethics committee take a decision based on the latest finding, latest research, and also latest outcome which is there. Another thing, last slide, don't write animal experiment which are like politically correct, but write something which is scientifically correct. Okay, great care, attention has gone, great care. This is political words. Great care don't means how many care. 1 hour, 2 hour, 1 day, 2 day. Attention has gone into refining the technique. Which technique? To monitor the animal response to treatment used. Which treatment? Anti-analgesic, anesthesia, or what? To reduce the degree and duration of suffering incurred to minimum work with the mice and cattle is not expected to be greater than moderate severity. Experienced observer, who observer? Experience. What is experienced observer? Whether they are veterinarian, trained to see that whether pain is there or not, and all these things. With access to veterinary advice and care at all times. Monitor clinical signs. Which are the signs you have to monitor? And all experimental regular interval. Regular means 24 hour, 2 days, 1 day, 1 hour, 15 minutes. What is the regular interval? And in order to identify quickly at animal require treatment, animal failing to respond to treatment is killed humanely. What is humanly? You're killing by thug, or you are killing by anesthesia. How you are killing. So you see, in this paragraph, everything is written politically. You don't know what is the intervention, you don't know the frequency, you don't know the duration, you don't know the adverse effect, you know little bit severity, and you also know pain. But not political words, but scientifically correct. What is the good example? Good example is this. Basis of experimental surgery is closure of the middle cerebral artery. So they are very clear. They are doing middle cerebral. What they were talking, great care. No great care. Under anesthesia. So they are clear. They are using anesthesia. This result in mice in stroke. Means mice will have a paralytic effect. Half of the body will not work, which manifests itself in hemiplegia, which constitutes the main factor and is classified as medium severity. Food intake is still possible. Animal will take food. That does not mean animal is not suffering. Animal still feel pain. Observation time is 90% of the animal for 24 hours. So first 24 hours, I will see 90%. I will be sitting with the animal, and rest on 10%. After 72 hours, 10%. Pain plays a central role as pre 6 to 8 hours post-operatively, and analgesics are administered. So you hear, it is not politics, not great care, not humane, nothing. You are crystallizing everything. You know intervention, you know frequency, you know duration, you have the adverse effect, you know the severity, and you know the fate. Everything is known. Nothing like humanely, great care, experienced observer, no. Here, everything is defined. So I wish that you write your proposal which are like this. Thank you very much for the attention, my dear friend. And I feel very happy. I feel very good that Professor Astuti and all of you colleagues listened to me and gave me the opportunity, at least virtually. I wish I could have come physically. If you would have been physically with us, then we would have a great fun. And also the experimental design. Thank you very much. I hope I have taken time in my little bit, I think, extra that I often take. Thank you very much. If you have any
questions. Thank you very much, Dr. Vijay Palsing. Let's make applause virtually to Dr. Vijay. by clicking the clapping hands on your laptop or your gadgets. Okay, I use my original hand then. Thank you so much for the remarkable presentation. Yes, there's lots, things that Dr. Vijay explained it to us, shared to us about the consideration how to use the animal in physiology, especially research, starting from the very beginning, how to decide which animal that we will use, how to prepare the animal, how to how to handle the animal, and how you get to the end with the accurate data and how to publish. I like the two statement of yours. One is the analogy, that how to do the checklist, by the analogy of flying from Jakarta to India. I would like to do that. To fly to your home, to your place. And also the last statement that when we write a publication about our research, not in political words, but in scientifically words. I like that. And I think all the participants here agree with me. Okay, do you mind, Dr. Vijay, to answer or respond if there is any question addressed to you? Okay, so I welcome participant who has a query or question they address to Dr. Vijay. Just raise your hand. I will check it, or someone, please help me to check if there is any question or participant that would like to address a question, because I don't have any in the chat box. Okay, this is from Reski Amalia. Okay, can you just directly address your question, Ibu Raski? Raski or Reski? Can you just address it directly?
Yeah.
Oh, sorry. This is Okay. Yeah, you're welcome.
This is the question from Dr. Reski Amelia.
Yep.
Good morning, Dr. Vijay Pal Singh. I permission to ask you why males still dominate animal study. Is the difference between male and female study? What is the difference? Very, very good question. And this is basically a big problem. Most of the time when we sit in ethics committee, people generally say, I'm doing always with animal and this animal is always male. Why male? Because female come into the cycle, this estrus cycle, and because of this reason there is a lot of variation in our data. So why they prefer to use? Because estrus cycle induce a lot of difference in your data. But can we use only male? No. As for ARRIVE guideline, we have to use both male and female. As we can see, we are not doing research only for the male. We are doing for male and female. If you are doing only on male, you cannot generalize your data. You will only say, I have done this experiment for anti-diabetic only in male. Hence, my result will be applicable to whom? Not all, but only male. Remember that, my friend. So, generalization, I told you, internal validity, external validity. External validity, you are limiting to half of the population. In our, now we have only around 309 participants, and most of I can see are the ladies. So if you are doing only on the male, how can you generalize to the female? No. So usually why people prefer male? So that they can get good result. Why good result? Because the variation which is there because of the estrus cycle is not there. That's why people prefer to use always male. Is it right to use only male? No, it is not right to use male. You have to use the both sex. It is now very clear in our ARRIVE guideline, unless it is justified. How justified? Sometimes you have a disease which is only for female, like uterus or like this ovarian cancer. Then of course you have to use female, or certain like in male. Otherwise, for the conditions like diabetes, cancer, and all these, you have to use both sex. My dear friend, this is very good question and also very good take-home message. Please use both sex. It is not your, you can say that only for your good experimented design that I will only use male and I will use only female. No, you have to use both sex. Thank you very much, Doctor, for the question, Rasika.
Oh, thank you so much for the nice answer. Is there any question that we address to Dr. Vijay? We still have, we still have time. Yeah, you are welcome. Yeah, Dr. Vijay. We just wait. Yeah, okay. That is okay from Reski Amalia. Thank you, she said to you. Thank you for your answer. Okay.
Thank you.
Is there any question? Okay, there is any question? No. Okay, no. Maybe during waiting for maybe the next question, I just want to ask you, a short question. If we want to, make like a standardization, and with the, most of the publications come from the European or the abroad that have the condition that different from the tropical condition in Indonesia. What do you think? We can use the data for standardization, or we have to make our own? Thank you.
Yeah, in this one, Doctor, whenever we are doing research, we have to be very, very careful. It is not that if I am doing in Delhi, then I can have my own temperature environment. No, in paper I have to mention that these animals are maintained into this temperature, this humidity, with this food, with this water, and with these all condition, light condition, humidity condition, air cycle changes, everything. That is the basically purpose, Professor Hera Maheshwari, that I can reproduce into even Yogakarta. So research is basically international. Whatever condition that you provide have to be written in paper. So ultimately there are two guidelines, prepare guideline and arrive guideline. Whatever condition I am doing experiment, that has to be written into your paper also, so that if you want to do the same experiment in your country, you don't have to come to me. You know, this mice, so you know the strain of the mice, age of the mice, sex of the mice, weight of the mice, when experiment was done, how it was done, whether approved by Animal Ethics Committee, what was temperature, what was humidity, what was the feed, what was the health status. So everything is known, everything is reported in your paper, and you can reproduce the same. So that is the basically the idea. If it is done in Europe, if it is done in America, if it is done in Indonesia. One example. Now we have done the research on corona. We have corona vaccine, but these vaccines are discovered in India. Is this vaccine is applicable everywhere in the world? So when we are submitting this data to WHO for approval, we submit all the data. And this all data include all these animal experiment, that this we have done this, so that anybody can reproduce them. Otherwise, if we are not doing by this, what you call them, patent, we are publishing. So in publication also, we mention all these parameters, temperature, humidity, this, that, all checklists have to be mentioned. Suppose you say, I have done experiment only in the male, and I am your reviewer. I will say, why male? Okay, whether approved by ethics committee, whether the all these parameters were taken care. So these things are now coming up internationally, Professor Maheshwari, and we have to be our central point is reproducibility, real science. As I told you, we are doing for humanity, we are doing for animal. Hence, we have to be very careful and also report the same. Thank you very much for the nice question. Standardization is very, very important. Actually, if I am in a real mode, I will do and I do a good exercise, but I cannot do virtually that one, so that people understand what do you mean by standardization.
Okay, thank you so much. And I saw there is, another question to you.
Yeah. Yeah, this is from a doctor.
Yes, risky.
Risky. Dear
Yeah.
Vijaypal Singh, animal experiment, 3-point component. Do these 3 components have to be fulfilled in animal experiment? Yeah, this is again a very good question. And the question is basically from the first slide, that what are the 3 components? What do you call an animal experiment? That it is to be done on living and vertebrate. Suppose if you are doing Drosophila, by ethical point of view, this is not an animal experiment. Suppose if there is no stress or suffering, you are simply taking blood for health monitoring. This is not an animal experiment. And if you are doing animal experiment, simply I want to do PhD, and there is no question, there is no problem you are answering, that is also not an animal experiment. These 3, Dr. Risky, these 3 components have to be fulfilled, and these are the ethical requirement or legal requirement in most of the country. You cannot kill experiment, and you cannot do experiment which is not useful. It has to have some importance. Why are you doing? Because ultimately, suppose if this is not done, what will happen? You are doing research, then you are publishing research. Publication means it will go international, everywhere. If you have not done your experiment correctly, I will take your experiment outcome and I will make my hypothesis based on your data. So we are amplifying the mistakes. So these 3 components are basically ethical requirement, and these 3 requirements have to be fulfilled before our experiment start. Thank you very much, Dr. Risky. I hope I'm clear. And these 3 I can again repeat. These 3 are performance of living and vertebrate, cause some distress or suffering to animal, and should have certain purpose. But importance is, purpose is not your PhD or your master degree or your MSc or your MTech, but purpose is a big societal problem you are going to solve, or for animal experiment problem. Thank you very much.
Thank you, Dr. Vijay. There is, another question. Yeah, I think the last one, because we are limited by the time. There is from, okay, let's hang on. From
Mila Hayati.
Mila Hayati. From Mila Hayati.
Yeah. This is also very, very interesting question. Are all study using rat or mice reflected the human condition? No. Are these certain limitations considering the anatomy and physiology? Yes. Of humans and rat or mice are very different. Yes. So this is very, very good question and need a lot of debate. But I will answer into as short as possible. Are all study using rat or mice reflected the human condition? No. Sometimes they reflect part. Like suppose one example, you are doing a research on asthma. Asthma means you breathe like very heavily. Asthma means airway condition. Such condition are existing animal naturally, yes. Which animal? Guinea pig. But asthma is not only airway hyperresponsiveness or this breathing problem, but inflammation also. Is inflammation there in guinea pig? No. So not all condition altogether present in animal. So your question is, all study using rat or mice reflected the human condition? No. Part, small part they represent. Are there certain limitations considering the anatomy and physiology? Yes. I give an example. We are doing a lot of diabetic research on mouse. What is the site of glucose clearance in humans? Site of glucose clearance in humans is muscle. Means we exercise. If we have a diabetes, doctor tells us to do exercise. What is the site of glucose clearance in mouse? Liver, not muscles like humans. So the question is, not all represented, but few are represented. And we are taking small part and then clubbing and making a big reply. Like suppose if you are saying your question number 2, are these certain limitations of considering anatomy and physiology? Yes. It is because of this reason that we are doing clinical trial, phase 1, phase 2, phase 3. Okay. because all anatomy and physiology is not represented, but certains are represented. Are human and rare? Now your question is means very correct, but I hope I could answer. So all study using rat or mice reflected the human, not all, not 100%, part. Are there certain limitation considering the anatomy and physiology? Yes, there are conditions. Like suppose in rat there is no gallbladder. So if you are doing research for diet or for food, rat is not a good model. If you are saying, okay, I am doing research on asthma, are you using Vista rat? No. Which is the good model? Guinea pig is the good model. So they are, remember, we are calling them model. Model means not 100%. Some, and there is limitation. Yes, there is anatomical and physiological limitation that I have discussed. Like in mice, you have a glucose site clearance is liver. In humans, it is muscle. We are exercising, we are running, so that glucose can be used in diabetes. So physiologically differences are there. That is for us to understand and use that and keep in mind while planning experiment that they are not 100% similar, and we are representing small part of the disease, and then we are finding out. My dear friend, most of the drug and discovery and device in everything have come through the animal. So animal experiment work. But we have to plan using our brain very intelligibly. So my question to you is not all 100%, but part. We have to be smart enough to plan experiment like that. Thank you very much. This is a nice question, Dr. Milhaya. I hope I am clear. But we should debate more and more when we plan experiment, when choosing animal model. We should have a good debate and criticism, positive criticism in between us. Why it is good, why it is not good. Diabetes in mouse is good, but not good in 100% way, because mouse eats all the time. Human eat all the time. No, we have breakfast, lunch, and dinner. Mouse eat it all the time. So alpha, beta cells which are there for this insulin secretion are the same. No, they are not same. And the gastrointestinal are the same, not same. So by keeping these component, we should plan experiment, and we should also know the limitation of our experiment. That because of this reason, it is not 100% true. External validity is not 100% true, but part. Thank you very much.
Okay, thank you. Actually, we are about to run out of the time, but there is, a short question. Can you just give a short answer as well about using the female rats, compared to the male one?
Yeah, good question from Dr. Nea Kurningis. What are the points that should be considered in using female animal in order to avoid research bias? Ma'am, Dr. Professor Nia, you should not use only female unless it is scientifically justified. So my question to you is that why are you using female first? If it is something general research or for everybody, you should use both male and female. Female should be only used when we cannot use male, like for ovarian cancer, uterine issues, or something like that. And I would advise to use both sex if it is unless scientifically justified. And what are the point that you have to consider the research bias? So while housing, if you are using only the female, don't bring the male into the room. Otherwise, they will have the smell and they will feel disturbed. They will come into the cycle. They will come into the cycle very often. So don't. If you have to use only female, use only female because of your scientific justification. Don't house the animal which is male into the same house. That is our reason. But I would request not to use only female unless it is a pressing demand of using only female. Use both sex. Otherwise, your external validity will be 0. Thank you for the nice question, Dr. Nia Kurnian Singh.
Okay, thank you so much, Dr. Vijay Palsing. Because we are limited by the time. Thanks again for your remarkable presentation, your nice discussion, and hope to see you in India or in New Delhi or in Indonesia in reality, not only in virtual. Okay.
I love Indonesia. Please invite. I will come. I will.
Okay, of course. And I would like to visit you in New Delhi as well.
Yeah. You are welcome. I'm very good person. I will invite you. Professor Astuti know my mail address and WhatsApp. Just send me a mail and I will invite you. I'm doing very good research on animals. Thank you very much, Professor.
Thank you very much. I'll see you later. Bye.
Bye.
Okay, ladies and gentlemen, I think we need to move to the other agenda. We will have another invited speaker, Professor Toshiya Muranaka. His current position is a professor at the Department of Biotechnology, Graduate School of Engineering, Osaka University, Japan. His former education is from Kyoto University, and his research publication, the latest one, it is about the analysis of NADPH cytochrome P450 reductases from legume for heterologous production of triterpenoids in transgenic Saccharomyces cerevisiae. This is published in 2021, and you can find this publication in Frontiers in Plant Science. Okay, welcome to Professor Toshiya Muranaka. The time is yours. Yeah, give applause to Professor Toshiya Muranaka. Okay, please.
Okay, thank you for introduction. So let me share my slides. Can you find the slide?
Yes, of course.
Okay, thank you very much. My name is Toshiya Muranaka. First of all, thank you very much to the Professor Endang. He's a professor in the Gajah University. She introduced me to talk in my topics here. And also thank you very much to the Dr. Surameto Widianto to have a chance to talk my research work. Actually, COVID-19, as a previous speaker mentioned, that now at the worldwide, and actually in Japan, also yesterday, the PCR positive marked the highest number recorded yesterday. Still, we have to keep on, but we are doing our experiment, of course. So today's title is a redesign of the biosynthetic pathway of plant-specialized metabolites towards human health. So the field is a plant science, and especially at focus on the crop development by genome editing. Maybe almost all of you know that very new technology, genome editing. So we apply this genome editing technology for the crop development. Okay. Okay, so the guest number to 1 million. Oh, this is a number that the plants produce. So the so-called secondary metabolites, or recently called specialized metabolites. Because plants cannot move, animal can move. So that's why the plants can survive with various different conditions to survive. So plants are faced on that some enemy. So plants kill by using the chemicals, and also that plants introduce that many much more big man to kill some other enemies. So that's why the plants produce 1 million. That's a very large number of chemicals. So these chemicals also have bioactivity. So this kind of chemicals can be used for the medicines or the food additive of a human also. So my lab is interested in to arrange these chemicals. So the keyword is gene discovery. of these useful chemicals, and also synthetic biology, and also genome editing in plants. So these chemicals are sometimes the content in plants is very low and also restricted to very rare plants, so-called very rare plants. And also chemical synthesis are very difficult. So the solution to produce a useful chemical is to, once we identify the genes for producing these chemicals and introduce these genes into the suited host. So sometimes in the yeast cells, sometimes in the plant crops, and sometimes the algae. So this is called the metabolic engineering. Metabolic engineering also, or the synthetic biology. So using this technology, we can produce useful chemicals in the sustainable way. So the one good example is shown in the yeast cells. Metabolic engineering is. Maybe almost all of you know the name of artemisinin. Artemisinin is an anti-malaria drug, which is produced in the plant Artemisia annua. So this artemisinin is only produced in the Artemisia species. So the solution to produce the worldwide artemisinin is the once you understand the gene for producing artemisinin, and they introduce these genes into yeast cells. And so that Amyris, this is a venture company in the United States, they succeeded in producing this artemisinic acid, 25 grams per liter, and by using the chemical reaction, they succeeded in producing the artemisinin a very large amount. So this is one successful story. And our laboratory is also interested in the other chemicals called glycyrrhizin. You know the name of the glycyrrhizin? It's a natural sweetener and also has an anti-inflammatory or anti-nuclear or hepatoprotective agent. And there's also some report to this is anti-SARS and also anti-COVID-19. So this is a very useful and important chemicals, but it is very difficult to do chemical synthesis. So idea is that identify the genes for producing glycyrrhizin. We have already studied more than 15 years, and we succeeded in identify the key genes for producing the glycyrrhizic acid. This is a precursor of the glycyrrhizin. So this P450s, 212, this P450s, and introduce these genes into yeast cells. And we are succeeding in producing glycyrrhizic acid. And this is now R&D stage with the chemical companies. And from these are, these are two P450s, glycyrrhizic acid can be produced. And also, And also we'd like to produce that glycyrrhizin in plants. So metabolic engineering in plants is the next. So how we can produce make a transgenic plant? This is one example to get a transgenic soybean. Okay, by using the explant with the somatic embryo, and by using the plantish culture technology, we make a somatic embryo. And during this course, we introduce genes for interesting. And finally, we can produce this one, transgenic soybean. Okay. So now we already succeeded in the all genes for glycyrrhizin. This CSYGT very recently succeeded in published in 2020 in the Nature Communications, and this one also, and also we can obtain these 4 important key genes for producing glycyrrhizin. And so the licorice plants and also the soybean in the same family. So our idea is introduce these genes into the soybean cells and produce and so the okay, sorry. So soybean which produce a glycyrrhizin. So this is now ongoing with a private company to produce the sweet soybean by introducing these important genes. So as mentioned, these are plant special metabolites as a very good use for the antiviral, anti-cancer, anti-malaria drug, and also sweet tasting in the case of glutamic, and the ginsenoside, anti-hepatic or anti-inflammatory. But some specialized metabolic is not so good. For example, for this alpha-solanin, it's a poisoning and also bitter taste. So this kind of chemicals can be eliminated from the crop, crops which we eat. So our idea is that once identified these chemicals, the solanin, and eliminate from potato by using genome editing. So today I focus on how we can identify these solanin biosynthesis pathway genes, and also how we can eliminate the solanin-less potato by using genome editing. Okay, so I already have a 20 minutes video. So please watch this video first, and then after that I can follow up. Okay. Just a moment. Potato, Solanum tuberosum, is a solanaceous plant. So it is one of the most cultivated crops in the world. However, the toxic compounds, steroidal glycoalkaloids, are highly produced in its sprouts and green tubers. So when large amount of SGAs cause food poisoning in human beings. SGAs are biosynthesized from cholesterol. This slide shows the biosynthesis pathway of phytosterol input in plants. In plants, most of the phytosterols are C24 alkali sterols, including cytosterol, stigmasterol, campesterol, and brashinolide. It's a phyto hormone. So generally, cholesterol contents are very low. In solanaceous plants, cholesterol content are higher than other plant species. This has long been mysterious why the solanaceous plants contain a high amount of cholesterol. In 2014, our collaborator, Dr. Sawai Riken, identified SSR2 gene, sterol side chain reductase 2, which catalyzes these reactions, namely cycloaltanol to cycloaltanol and desmosterol to cholesterol. So SSR2 is first duplicated the common SSR1 gene and then evolved to the SSR2 gene. SSR2 genes are only found in solanaceous plants. That's why cholesterol content and downstream steroidal glycoalkaloids are high in solanaceous plants. So if we could disrupt SSR2 genes specifically, we could obtain potato plants with reduced SGA level. However, so far, it has been almost impossible to disrupt the targeted gene in plants. Now we can disrupt the desired genes by using artificial nucleases. This system is called genome editing. Site-specific nuclease such as zinc finger nuclease, ZFN, and transcription activator-like effector nucleases, TRNs, and clustered regulatory interspace short palindrome repeat associated protein 9, CRISPR-Cas9 system, recognize a specifically sequence in the genome and introduce DNA double-strand break, called a DSB. So this is the case of the TRNs. When two monomeric TRNs are designed as shown here, and FOX1 domain will dimerize, and shown in the nuclease activity in the specific locus within the genome. activate the FOC1 domain, FOC1 dimer will induce the DNA double strand break between two TARN binding sites. So after double-stranded DNA breakdowns were introduced to TARN, they were repaired by the endogenous system called non-homologous end joining. So through a non-homologous end joining system, double-strand DNA will be repaired incorrectly. So we can introduce target mutation in the TARN target site within the transgenic. And also using the homologous recombination, we could knock in the origin. When we started our project to disrupt the SSR2 gene, CRISPR-Cas9 system had not been developed yet. So that's why we applied the TARN technology to disrupt the SSR2 gene in potato. As I mentioned, because SSR2 first duplicated in the SSR1 and evolved, both two genes are highly homologous. So it was challenging whether our designed TARN module really target SSR2 and no effect to SSR1. This slide shows the transformation of the potato with a TARN expression vector. Once we have obtained the TARN binary expression vector, and this vector is introduced to Agrobacterium tumefaciens. And using the Agrobacterium mediated transformation, we infected the microtubule or leaf discs of potato, and the screening by the selection by the kanamycin resistant plant. And we obtained the independent transformant. And the PCR amplified across the target site of the genome DNA. And using heteroduplex mobile assay, we can fast screen the putative DNA, putative genome editing plant clones. Among fast screened potato clones by the heteroduplex mobile assay, clone 19 here has no intact sequence in the target sequence of SSR2 as shown here. For example, this means that 5 nucleotide deletion here for 3 times. And here means that 25 nucleotide deletion and 8 nucleotide insertion. So we then analyze SGA contents in the transformers and found that both the storanin and the chaconin are dramatically reduced in the clone 19, as shown here. So potato crops are highly heterozygous tetraploid. So this is the first study demonstrating metabolic regulation has been done by genome editing. However, a problem remains. Potatoes are, as shown as tetraploids. So therefore, if this clone is homogeneous, the sequence pattern should be less than 4. However, as shown here, there are 9 patterns. So this suggests that this clone could be a mosaic. So highly active TARNs with variable TAR repeater harboring non-RBD variation called platina TARNs have been developed and first applied to human cells. So we are applying this platina TARN vector for the plant to design and target the SSR2. Was usually constructed and introduced in the potato plants. So we obtained the candidate clones for the genome editing in SSR2. So we checked that whether mosaic or not by analyzing different positions of leaves. And these 3 clones were selected. So we sequenced the target site of these clones, namely the number 11, 71, and number 186. And these 3 transgenic lines, at least 15 random selected clones are shown here. So we could not detect their intact SSR2 sequence. So almost all the detected mutants of the clone was a simple deletion. Well, when compared with the sequence of the SSR2 gene. And there was less than 4 of those patterns. And also, based on the direct sequence analysis, we observed no mutation in SSR1 in all the 3 transgenic lines. Here's data not shown. So SGA contents of these 3 clones were, as shown here, dramatically reduced compared with those of the wild type. Furthermore, they showed normal phenotype as far as pot cultivation were done. So in this case, the cultivar is the name is Sashi, and we have made a publication last year. So as shown here, the normal tubers of genome editing potato were successfully obtained. By using platinum, we succeeded in disrupting all 4 alleles of SSR2 in tetraploid potato plants. Furthermore, there was no off-target effect to SSR1, which is very similar to SSR2. For plant genome editing, the genome editing reagents are usually expressed in plant cells from stable integrated transgene within genome. In this case, a TARENs here. So this requires crossing process to remove foreign nucleotide from the genome to generate null segregants. However, in highly heterozygous plants such as potato, the progeny lines have different agronomic traits from the parent cultivars and do not necessarily become elite lines. So how we can obtain null segregant in heterologous tetraploid potato without changing their elite genetic lines? Thus, after infection of Agrobacterium, genes in tDNA region are introduced into plant cells and finally integrated to the plant genome. In this case, a wild-type agro, Krangol is obtained. In fact, after Agrobacterium infection to the plants, tDNA are introduced into the plant cell. It's finally inserted in the genome. The gene on tDNA is transiently and strongly expressed before integration. So thus we expected a strong transient expression of the gene before the genome editing cassette corresponding to the scissor was inserted into the nucleic genome. So this is an attempt to edit the target gene by producing a lot of scissors in this transient expression. So how we could obtain that genome editing plants without integrating into the genome? So the idea is very simple. Usually we screen transgenic plants by kanamycin or something other antibiotics to obtain the transgenic plants. So in this case, we don't do selection. So we obtain the many regenerated plants and the subculture, and after that, HME sequence analysis, and we screen that putative genome editing plants without integrated in the plant genome. So in the screening, after the HMEA screening, we analyze the candidate clones and finally selected these 7 clones. As shown here, the number 117 and number 75 show the no intact sequence of target SSR2 genes. And SGA contents are analyzed in these 7 clones. Among these clones, number 117 and number 75 are no intact SSR2 sequence showed dramatically reduced SGA contents, which means that we have obtained 371 regenerated plants among these 2, which means that this one and this one of regenerated shoots don't have intact SSR2 with no detectable transgenic integration, which means about 0.5% plants can obtain over this method. So now we are planning to field test from this year. However, because of the COVID-19, still we have to wait. So anyhow, using this agro-infection system, we can obtain the genome editing plants without integrate into the plant genome. Not only potato, but this technology can apply to the many other vegetative and also polyploid plants. So thus, we obtained the regenerate lines with disrupted SSR2 gene and without the transgene of the target genes in the genome. So next, we have to consider this type of genome editing plants are regarded as GMO or non-GM. To use genome editing organisms properly, it is necessary to establish handling rules. Internationally, genetically modified organisms are regulated by the Cartagena Protocol on Biosafety. In this act says, genetically modified organisms and others refers to an organism having a nucleic acid obtained by use of the following technology or copy thereof. So, GMO is defined as the organism containing extracellular processed nucleic acid or its replicate. In the last part, I'd like to briefly introduce the recent regulation framework of genome editing organisms and foods in Japan. In Japan, Ministry of Environment, MOE, has a responsibility on the Cartagena Domestic Act. And the genetically modified foods and feeds are regulated by the Food Sanitary Act by Ministry of Health, Labor and Welfare, MHLW, and also Feed Safety, Ministry of Agriculture, Forestry and Fisheries, MAFF, respectively. So, handling policy has become necessary for the social implementation of genome editing organization. In the last year, in late last year, in late 2019, MOE formulated a policy for handling genome edited, and MHLW established a policy for handling genome editing food and food additives. So this figure shows a category of genome editing. Genome editing is defined as shown here, namely SDN1, SDN2, and SDN3. SDN means a site-directed nuclease. So SDN1 means that it's a site-directed nuclease 1. In this case, targeted mutagenesis occurs, deletion, insertion, substitution by a non-homologous end joining. This non-homologous end joining is a naturally occurring mutagenesis. So this type 2, gene targeting, SDN2, is a substitution of several bases, or SDN3 means a foreign gene integration by homologous recombination, HR, using template. So this type of the gene targeting is not so often happens naturally. According to MOE handling policy shown in last year, when genome editing organism is categorized in SDN1, which means only deletion, insertion, substitution, and it does not contain a foreign DNA sequence, which means a null segregant, the organism may not need to be regulated as a genetic recombinant if notified to the regulatory authorities. So in our case, SSR2 disrupted potato with agro-infection will be regarded as out of the scope of GMO regulation. Similarly, the MHLW last year has indicated that genome editing foods and food additives may not need to be regulated as genetically modified food if they are derived from the null segregants. And the modified DNA sequence is within the range of natural mutation. So again, in our case, SSR2 disrupted potato with agroinfection will be regarded as out of scope of GMO regulation when we could show the evidences described here. So this is the case of Japan. We need more arguments and also evidences whether these concepts shown here in Japan will be world standard or not. And in the video, as I mentioned, we are planning to field test this last year, but because of the COVID-19, we could not start. But now as shown here, okay. Yes, from this year, very, very recently, this April, we could start the field test for the, not for the selling to the market, but still the scientific study level. But we started a field test at Naro. It is located in Tsukuba under the next regulation. And we started potato. And we are very interested in what kind of potato we can obtain this late summer. Yes, our potato is still experimental stage. However, in the tomato, it is called a GABA-rich tomato. GABA-rich tomato has been already invented, breeded by the genome editing technology. In this case, using the CRISPR-Cas in the Tsukuba University people, and also the joint venture company of the Tsukuba University, the Sanatex. And they are already improved for the this one, the Food Sanitary Act, Cartagena Act, yes, and then Food Sanitary Act, not to regulate as GM, but out of the scope of GM in the last year. And that's why this potato is now, we can get, still we cannot get in the market, but they deliver the volunteers who would like to cultivate in the house. So free distribution of seedling has been started last month. And actually, I'm also plant this GABA-rich genome editing tomato in my house. And I'd like to waiting for taste this genome editing crop, genome editing vegetables in the world, first in the world. Okay, thank you very much. I think you enjoyed the slides, but actually some, script is a little bit old because this script has been done 2 years and 1 year before. So let me follow up a little bit. Okay, this one. Yes, our, SGA, poisonless potato, the field test has been done last year, 2021. It's still the experimentary field test, but we are now testing the agricultural phenotype trait for this season. Next season, we also try. And also GABA-rich tomato. GABA is a gamma aminolevylic acid, which tomato has been already, served in the volunteers from that North Hokkaido to Okinawa South. And also this, non-GM genome edited tomato has been available all over the Japan. And also we can buy this GABA-rich tomato. And as for the experimental stage, our potato and also the rice and wheat field test has been done. Is now ongoing. So now from the 2019, sorry in the Japanese, but we are focused on the various the genome editing crops, applied genome editing crops for the potato, wheat, and ornamental trees, and piman, and rice, and soybean, and radish, and also onion, like this. And also established the Japanese-made genome editing technology also. So now I don't know, still don't know that still our idea for the non-GM genome editing law will be worldwide or not. But I think this is a very logical way. And I'd like to acknowledge these guys, especially the Dr. Umemoto and Mizutani, who study more than 15 years for the developed transgenic, genome editing potato works. And also, I'd like to make more 2 minutes to introduce our recent idea for that food loss and waste innovation hub by Osaka University. This hub has been launched last year, and we are focused on the food loss. And especially for the food loss is a various, type of production loss, storage loss, processing loss, transportation loss, consuming loss, and also intake loss, this kind of things. So we focus on these 5 major causes from that using the biological and also technical or social science, various technology field. And also we focus on this, food loss problem with high collaborate Indonesian people, Indonesian scientists. We have a very strong connection with Indonesia, especially Professor Fukusaki. He's the head of this research work, has a very good connection with the Indonesia, a scientist, and also very interested in the Indonesian tropical foods. So these are members of the players of the hub. So who are interested in, please click this QR or please send an email to me. And I also the member, and I focus on this food loss by using the genome editing technology. Okay, so thank you very much. And yes, this is the member in the 2019, 2 years before members. The reason why I use this picture is we have various international students, especially the Indonesian, Thai, and also the Vietnam, and actually here Agus, he is from the Gajah University, and also Fanani is from the Airanga. So these 2 guys already finished a PhD course. And now that these members still have a mask, we are not to stay home, but we are doing our experiment. Okay, thank you very much.
Okay, thank you very much, Professor Muranaka, for your awesome presentation. Yeah, you have, give us, a very important and interesting insight about genome editing, especially to construct metabolites in plant. I know it's a very difficult, and you give us explanation from the very beginning, how to discover the gene, how to synthesize, and how to make a genome editing. I think there is a lot of thing that we can use, for medicine, for the GMO, for the healthy food, maybe. Yeah. Okay. This is very interesting, and I welcome to all the participants who will address a question to Professor Muranaka. Please, we still have time. Okay, you can do by raising your hand or writing your question in a chat box. Yeah, you are welcome. Maybe especially to whom from plant physiology, or to whom who currently working in the area of molecular biology and genomic is very interesting. Okay, this is from Nia Kurnianingsih. Okay, should I read it for you, Professor Marunaka? Okay, this is a question from Nia Kurnia Ningsi. A very interesting presentation, Prof. I wonder if genome editing is applicable to biofortified major staple food such as rice.
Yes, okay, thank you very much. Very important questions. Yes, actually, we can apply these generating technology. Many are various crop, including rice, of course. And in Japan also, we are doing the genome editing technology application for the rice plants to high productivity, or changing the day period or flowering time or something. So various technology has been applied for the rice using genome editing. Also, Tokyo University people are now starting also the field test, experimental field test of genome editing rice from last year. Okay. And the other question is
thank you.
Application for that livestock animal feed. Yes, of course.
Mm-hmm.
And yes, very good example is using the GM. In this case, GM some herbicide resistant animal feed, or disease resistant has been done for the GM technology. But as you know, GM is sometimes very highly restricted. So our idea is this kind of disease resistant or insect resistance, or sometimes the herbicide resistance can be do by using the genome editing technology. So in this case, it is not regarded as GM. So it is more friendly, acceptable for that for the people. So this kind of study is also done in the various in the world.
Okay, thank you, Professor Muranaka. Is there any question? Okay, during waiting for the next question, maybe I will ask you some, a short question, Professor Muranaka. How about the, the potential of using like potential metabolic or secondary metabolite from the plant for medicine in human, like a traditional medicine? What do you think about that?
Yes, of course, this is very important to apply for the traditional medicine, because sometimes the traditional medicine means that medicinal plants can be found in the very restricted region. So, but by using our metabolic engineering technology, we don't have to cut. We don't have to obtain from the wild plants, but we can, once we can understand the genes for producing secondary metabolites, we can introduce, as I mentioned, that the cells or some crop cells, and we can make these are used for chemicals sustainably. So this is also one. It is also environmentally friendly and sustainable production. So there's a various application for the production of the chemicals which are traditional medicine is used.
Okay, thank you very much. It's interesting. And also, I think your offer to collaborate with your team in your laboratory is, thank you very much. Yeah, maybe anyone wants to create or want to, start a collaboration, research collaboration with Professor Toshiya Muranaka. Yeah, is it possible? Yeah.
Of course.
Yeah.
Please contact me.
Oh, yeah.
Okay.
between institution. This is good. Okay, is there any question? Okay, we still have some more. Okay, there is a question from Reski Amalia. Very interesting information. How long? Okay. Okay. How long does the gene editing process take to succeed? Is there a certain time limit?
You mean that how long does it take to R&D the genome editing crops? So I guess it's a very good question. Actually, it's depend on the crop. I mean that usually this genome editing crops is using the usual tissue culture technology. So if the tissue culture of the target plants is very easy plants, so it is very short time. But sometimes that very difficult plants for tissue culture, it takes time. And also that some tree is very difficult because it takes time. So it depends. But in the case of the rice plants, using maybe that 1 year or 2 years, we can get a first prototype of the genome editing crops. Okay, thank you very much.
Okay, so there is no a certain time or period, that we can say for the processing of genome editing, because there is a lot of maybe a problem or maybe a process that we have to carry out. Okay, thank you. Okay, any other question? We still have Professor Muranaka here. This is a good opportunity for you all to share, knowledge. Maybe you have, experience that related to Professor Muranaka research or topic. We can share, or you can ask any other question. Okay, from Mila Hayati. How does genetic modification affect the therapeutic efficacy? How does genetic modification affect the therapeutic efficacy of the plant?
Affect the therapeutic
efficacy of the plant.
You mean that therapeutic efficacy to plants? I don't know the meaning of the question.
So it's mean I don't know, Dr. Mila Hayati. This is about the disease in a plant, the problem in plant, or what? The efficacy of the plant, the therapeutic efficacy of the plant. Plant as an object or plant as a subject in here?
So for example, we make for example, the anti-disease genes are introduced, and the ones one transgenic plants can obtain, we can get a antifungal or insect plants can be obtained. So is it okay? So it depends on what genes we can manipulate. Depend on that what gene you manipulate.
Okay. Is it answer your question? Dr. Mila Hayati Dole?
Okay. Okay. Hi.
Okay. Thank you, Professor Muranaka.
Hi.
Okay, is there any? We still have time, I think.
No.
I have a question, Dr. Heran.
Yeah, you're welcome.
Yeah. Okay, thank you. Nice presentation, Prof. Muranaka. It's according to the genetic modification, yeah, that your study. Is there probably effect on the health, our health condition related with the genetic modification of the just like rice or others plants that we eat in every day? Okay, thank you, Morana.
Okay, and I understand what you mean, sir. Yes, GM is usually GM is exogenous genes are introduced into the plants, and this is normally occurred. So that's why the people is very anxious. That's why the many tests to secure the risk has been done. So many are tests for that for the human being and for the safety. So now that the GM, which has been preparated worldwide, is how to say very safe, because many kind of the safety test has been done. So, and also the GM, not the GM, but genome editing crops. It is very brand new technology, but this is brand new, but very how to say the one kind of the usual crop breeding system, because what the genome editing plants is that also occur in the natural natural plant breeding system. So now we can we can define that GM and the non-GM but genome editing. So that genome editings are more friendly, acceptable for the human beings. So I think so. Okay.
Thank you for your question. Yeah.
Okay, thank you, Dr. Diki. Professor Muraka, from the previous question, there is any additional information about the effect of the genetic modification to the efficacy of the plant? Yeah, this is actually the meaning is the efficacy of a therapeutic efficacy of the plant that use as a medicine. So because the genome editing or genetic modification, yeah, is used as well for the plant development, for the crop development, and what actually the effect for the therapeutic efficacy if the plant used for the medicine.
Yes, as I mentioned, it depends on what gene manipulated.
Mm-hmm.
So it depends on what gene you manipulate.
Okay. So it's depend, right?
Yes, depends.
Okay. Yeah. But I mean, the effect is usually, or is always positive, or is there any maybe negative, or maybe the efficacy of the therapeutic maybe will decrease?
I mean that if you'd like to obtain that fungi resistant plants,
Mm-hmm.
okay, so we introduced the genes
Mm-hmm.
for that fungi resistance. So this plant is very normal if that fungi is infected. So in this case, it is a very higher therapeutically good effect for the plants. So I mean that it depends on the plants. It depends on the genes which you introduce or change.
Okay, so there is no, I say like a not really bad actually effect in the process to the secondary metabolism of the plant, I think. No?
Yeah.
Yeah.
So we can, how to say, introduce that, the content of secondary metabolites can apply for the drug also. Yeah. And also, as I mentioned, in the case of the potato, we'd like to eliminate the poison. So we poisonous chemical, then we can knock out the gene for producing that poison SGS. Now we can make that the poison-free potato. It's very good for the human being.
Okay, thank you. Hope this answer will answer your question. Okay, thank you so much, Professor Muranaka. Okay. Yes. Thank you, Mila Hayati. Thank you so much. Hope it's answer your question. Yeah, Vincentia, Dr. Vincentia helped me as well to interpret more clear your question.
Okay.
Okay, is there any still welcome for the next question? Maybe anyone want to propose any collaboration, research collaboration with Professor Muranaka? Anyone want to propose in this event, in this time? Okay, if not, I thank you again, Professor Muranaka, for your very nice presentation. I hope we can, see again in other event, maybe in Japan or in Indonesia. Okay, thanks again. Please give, applause to Professor Muranaka virtually only. Okay, thank you. Thank you for, yes, yeah, you have, a clapping hand. Okay, thank you again, Prof. Muranaka. Okay. Okay, ladies and gentlemen, we come up to the last presentation of our session today. We have the third invited speaker. There is Dr. Diki Muhammad Rizal. Master of Health, yeah. Okay, Dr. Diki current position is at Department of Physiology, Faculty of Medicine, Public Health and Nursing, University or Gajah Mada University, Yogyakarta, Indonesia. His former education, medical degree from Faculty of Medicine, Diponegoro University, Semarang, and that's it. And his master degree from Biomedical and Reproduction Sciences program, postgraduate medicine, University Diponegoro Semarang as well, and andrology specialist from Collegium Andrologi Indonesia, and basic education and continuing sexology sciences from Faculty of Medicine Udayana, and PhD degree from Faculty of Medicine Gajah Mada University. His research publication, the recent one, the latest one, is about adjustment of sperm cryopreservation laboratory management during the COVID-19 pandemic. And you can find his publication in Journal of the Medical Sciences. Okay, so I welcome Dr. Diki to have your presentation. Time is yours.
Thank you, Dr. Hera, for the introduction. Yeah. Assalamualaikum warahmatullahi wabarakatuh. First of all, I will introduce myself. My name is Diki Muhammad Rizal. I'm a lecturer in Department of Physiology, yeah, especially in Physiology and Reproductive System. I'm also as a clinical andrologist. So this topic is very relevant, has a high relevance with my field of study or my clinical study, yeah, especially in my profession as a specialist in clinical andrologist. Okay, thank you for the committee, yeah, that the honors or for the times invite me as a speaker, yeah, as a keynote speaker. But maybe this is not the last time for we held the symposium, because of this is the third symposium, yeah, the third international symposium of global physiology. Okay, I will start my presentation, especially with the topic of or the title of impact of COVID-19 on male reproductive problems. Yeah. Okay. I will share my presentation. Okay. You have seen my presentation, Dr. Hera?
Yeah, exactly. Yeah,
Okay.
it's clear.
Okay. Yeah. Okay. I give that my I give a subtitle, yeah, in my presentation is what do we want to know and care, yeah, because of especially for the people said that COVID-19 is pulmonary problems or respiratory tract problem, but we know that multidimensional of multiplier effect of the COVID-19 in our body, and so until the site is male reproductive, yeah. So we know now we concern in male reproductive system. Yeah. Okay, the word was shocked at December 19, yeah, at the Wuhan, China. There is the condition, that a horrible condition, that so many peoples, yeah, so many peoples suffered the mysterious disease, yeah, mysterious disease. And then the government take a decision to lock down, yeah, and so many activities it's going to stop, yeah. It's very horrible condition, and then spread to the world, yeah, the other countries, yeah. And then WHO state that in March 2020 that the world is going to the pandemic situation. Now, this is the era that the early era of COVID-19 pandemic, yeah. And also in Indonesia, yeah, in the middle of March, yeah, we have a first patient of COVID-19. Yeah. COVID-19 is a viral disease. Yeah. That the COVID is coming from the terms coronavirus disease. 19 maybe due to the times, yeah, that for the first time emerged in the China, in Wuhan. Now, COVID is the etiology of COVID is caused by the virus with the name is SARS-CoV-2. Yeah, SARS, it means severe acute respiratory syndrome. Yeah. And CoV-2 is coronavirus 2. Yeah. Just clearly that for the first time, the focus of the COVID-19 or coronavirus disease is a pulmonary disease or respiratory tract infection by the virus. It's clear, but and then after the times, and then the researchers showed that so many problems with the patient with the COVID. The researchers find that COVID have not only in pulmonary side and also in extra pulmonary side. This virus is very, very seriously because of with the impact of mortality, dead people by the attack of virus or COVID, and then the increase the dead people with the COVID-19. But the impact on health, you can see that there are two kind of impact on health. For the first is asymptomatic, and then the second one is symptomatic or with the illness, with the sign and symptoms. This is the appearance of the COVID-19, or SARS-CoV. That is a corona, with a spike here. A bit beautiful, but it's so horrible. Okay. What about the situation now? At the present, I think 19 January 22. It's about the 59 million people currently infected by the COVID. It's very, very high number of the disease because of this is a pandemic. But now it's declining of the serious or critical. And it's very decline, very decline of the serious or mortality in so many countries. And so there is a COVID, but it's more maybe it's more likely mild or until moderate of COVID. And then the recovered is so high, 80%. But still the problems because it causes the deaths or mortality. 5.5 million people die because of the COVID. What about the Indonesia? In Indonesia, it's just the same. Indonesia is with the big population. I think it's more clearly that Indonesia is also has a big number patient of suffered of COVID. Maybe this data showed that data tell that 4.2 million people. And then 144,000 of the deaths. It's so high. It was a shockly, and so it's the government will take a big decision, to combat the COVID-19. Okay, until February 2021, it's still 4 variants. It's still 4 variants: Alpha, Beta, Gamma, and Delta. But now, at the present, the world is come to us the fifth variants, we call with the Omicron, with specifically it's more light of sign and symptom, but we think that it's still mysteriously, still mysterious because of this variant. It's it's still under research. I think it's under research. But the Delta is very make increasing of the deaths. So many people deaths by the Delta variant. Okay. As I said before, that COVID has a target extrapulmonary and pulmonary. We can see that COVID-19 can attack in the intestinal and also in the into cumentum and skin, and then in the renals, and then eye, We know that in the first time COVID emerged in Indonesia, there is so many symptoms that related with the eye complaint or eye symptom, just like conjunctivitis, It's a very common of the sign on symptom of the COVID, and then from the cardiovascular also as a target of COVID-19, and then this is a part of the symptom, myalgia, myopathy, and neuralgia, and also as a topic, my topic presentation is a male reproductive system. So we know that COVID has multiple organ as a targets. So it's maybe it's not easier to say that COVID only with the sign of symptom respiratory tract, but we must maybe a lot of cares of the others sign and symptom or the others effect on the others organs. Before we talk about the COVID impact on male reproductive system, I will let you to reminding about the male reproductive system. We know that the male reproductive system, classified into the big four. What the big four? For the first is a testicle. We have two testicles. The men have two testicles. With the function, the main function is spermatogenesis and steroidogenesis. Spermatogenesis, it means produce the sperm, and then for the steroidogenesis, produce the androgen hormones, especially testosterone. And then proceed to the second one is male tract, male reproductive tract, consists of epididymis and then vas deferens and also urethra. Epididymis, it's very important for the male reproductive function because of in epididymis there is a continuation of maturation after the maturation at the testis. This is the first maturation. It's about the morphology. The sperm have a tail, sperm have a midpiece, and the sperm have a head. It's in or within the testicle. After this, or it's in epididymis, the sperm will be gain of the motility ability. It's very wonderful site that the epididymis is a very important, especially for the capability of fertilization process. And so if there is the problem in epididymis. Yeah, there's the big question: what about the sperm function, especially of motility? Yeah, and then the next male reproductive tract is a fast difference. Yeah, fast difference is the way for the sperm transport, and then urethra. Yeah, urethra is also the sperm transport of the sperm tract. The sperm expelled from the body. And then the third one is male accessories gland. Yeah, there is a pulpo-urethralis gland, and also the big two, yeah, prostate gland, yeah, and seminal vesicle. Seminal vesicle is the biggest glands, yeah, that contribute to 70 until 75% of seminal fluid, and the prostate gland produce about the 25% of the semen, yeah, semen fluid, yeah. Okay, because of the important rules of this glands, maybe without the glands, the nutrition of the sperm will be decreased and then interfere with the disturbance, especially for the motility. Yeah, and the last is a penis. Penis has the function for the copulatory, yeah, copulatory, and then have a role in the transport of the sperm. Yeah, it's clear that the four big organs in male reproductive system that I said in this slide. Yeah. Okay, in deep on the testicle, yeah, within the testicle there is a tubulus seminiferus. Yeah, tubulus seminiferus. Tubulus seminiferus is a place for the spermatogenesis process. Yeah, from the spermatogonium and then spermatocyte, spermatid, and then the mature is a spermatozoa. Now, there is the important thing of blood testis barrier, blood barrier of testis, yeah, blood testis barrier is a Sertoli cell here. Sertoli cells is a part of the tubulus seminiferus and as known as substangular cells. Yeah, it's very important because make a defense mechanism for the spermatogenesis process from the gametes. Yeah, but after the gametes of the Sertoli cells, and then the blood testis barrier was broken, yeah, it will lead to increasing of the produce of antibody, yeah, that will attack of the other sperms because of the sperm will known as antigen. This is a response, immunology response from the broken of blood testis barrier, including from the infection of testicle, yeah, also by the virus. You know that the parotitis, yeah, is mum virus, is also give the problems in male reproductive, especially not only the swollen of testis, it's also the problems, the produce of antibody in the sperm, yeah, antibody sperm, yeah. It will reduce of the fertility or fertilization capability. Yeah. Okay, this is the part of the testicle. Yeah. For the activity of male reproductive, yeah, there are 2 points of male reproductive activity. We know that we call it the procreation and then the recreation. For the procreation, and I said before that there are 2 process, spermatogenesis process lead to produce the sperm, and then steroidogenesis process lead to produce of hormones, especially testosterone, androgen hormone, especially testosterone. What about the sexual activity? In sexual activity, especially the organs that involved in sexual activity, in the function of the organ is penis, and then male reproductive tract, and also male accessories glands. Yeah. What about the testicle? Testicle is not fully involved in the sexual activity because of testicle has released the testosterone, yeah, during the sexual activity. Only the testosterone released during the sexual activity. Nah, what the male reproductive problems? Yeah, okay. The male reproductive problems consist of the first is the big number is infertility problems. Yeah, and then the second one is infection problems. Yeah, okay. COVID, coronavirus, viral disease is infection problems. Yeah. And also, if the testicle was suffered by the COVID, it's due to for the condition we call with the orchitis, yeah, the inflammation process in testicle, yeah. And then the third one is sexual dysfunction or sexual problems, yeah. Maybe you can see that there are several sexual dysfunction problems, just like erectile problem, yeah, erection problem, and then desires problem or ejaculation problems and orgasm problems. Yeah. And what about the fourth one is a congenital anomaly. We know that in some cases, there's the child with the inhibition or the late of the growth of the genital. We call it the hypogonadism. Yeah. And then what about the malignancy? The last problem is a little bit of malignancy in male reproductive system. Yeah. Especially is testicular cancer. Yeah. Testicular cancer. Yeah. Maybe in the form of seminoma cancer or seminoma testis. Yeah. Now, there is an interrelationship between the infection, hypogonadism, or malignancy, and sexual dysfunction due to the big one problem is infertility. Infection problems can cause male infertility. Hypogonadism with the low of testosterone, yeah, it had an effect in sperm motion, yeah, or sperm motility. Malignancy is also give the problems may be related with the decreasing of healthy tissues, yeah, and then decreasing of the sperm numbers production. Also give the problems related with the medication, yeah. And the sexual dysfunction, as we know, in definition of infertility, there is a definition about the frequency of sexual activity, yeah, 2 until 3 times in year without the pregnancy, yeah. And so sexual dysfunction give the contribution for the infertility, male infertility problems. Yeah. Okay, this is about the infertility because of it is a big problem in male reproductive. Yeah, time of marriage, frequency of sexual intercourse, and without barrier contraception, and how to know about the fertility status. Yeah, we can or fertility status condition we can find by the sperm analysis with the manual WHO sperm analysis. And so I want to say that sexual dysfunction, it's a part of the male infertility problems. Yeah, beside the indeed in the sperm production or sperm quality. Now, for the male infertility problem is coming from the so many problems. Yeah, that maybe, as I said before, sexual problem, sexual dysfunction, and then seminal plasma abnormality and STD, and testicular damage. Yeah, testicular damage. And I said before about the broken of barrier testis. Yeah, blood testis barrier is the anti-sperm antibody. Yeah, is emerge or produce sperm antibodies. We call it the anti-sperm antibodies that make infertility problems. Okay, I will proceed to the pathogenesis of the COVID-19. Yeah. Okay, I'm not expert in this topic, yeah, because of I'm not a microbiologist, yeah, and or a pathological doctors, yeah. But I must give introduction before we entering to the problems in male reproductive. SARS-CoV, how to attack our body is coming from the interaction between the virus, yeah, between the virus and the receptors. These receptors, there is two of receptors, ACE2, yeah, angiotensin converting enzyme 2, and then the protein, yeah, protein serine, yeah, TMPRSS2, yeah. What about okay, next, yeah, I will try to explain. And then after this, yeah, after the S protein, yeah, will be attached to the surface of our cell membrane. It will be enveloped and then continue to the COVID, the SARS-CoV to the our body. Our body is a host of the infection. ACE2 physiologically, basically is a physiologically in our body. We know that angiotensin converting enzyme is an enzyme that very important for the maintenance of the blood pressure, our blood pressure. Yeah, and it's very important. But in this case, for the COVID-19 yeah, problems, the ACE2 receptors as a facilitator for the virus entering the body. Also, the TMPRSS2 yeah, has a biological effect in so many moments or so many process in our body. For the SARS-CoV-2 pathogenesis inside in our body is as a priming of S protein, yeah, and so before the envelope in our body, the activity of TMPRSS, it will continue to the entering this virus to our body. Okay, this is just like it's similar, yeah, similar with the last slide, but we know that the viral RNA is released, and then replication and transcription of the viral genome begin within in the host cell. After entering, it will be replication and transcription. And I said before that ACE2, yeah, it's very important in our body because of involved in maintaining of the blood pressure, yeah, homeostasis, maintaining of blood pressure with the function is very important, yeah, especially in vascular. Now, This is the expression of S2 receptors in so many organs, including the testicle, including testicle and prostate, including prostate, especially in prostate, the pancreas, kidney, male tissues, male tissues, testicle, and also female tissues here. What about the MPRRS expression? It's also the same, just the same with the ACE2. It's also so many places of so many organs in our body also express the protein of the MPRSS here. After the virus going inside in our body, there are two types of continuation. For the first, resolution infection, and then suppress inflammation and recovery. But if there is an unlucky, there is a progression of infection to severe inflammatory action and cytokine storm and ARDS. And so this is make a respiratory failure and then sepsis and the increasing of the rate of mortality. For the morbidity is sure, but of course, but the mortality is in some patient will be got the mortality or the death condition. What about the bisex? By the sex, there is increasing in male compared in female. This is I think it's suspicious of COVID.
Excuse me to interrupt. You still have 3 more minutes, Doctor.
Oh yeah, okay. With the sex, with the male sex, I'm going to proceed. After in testicle, there's so many after testicle, after inside the testicle, is so many problems, direct destruction, and also affect the Leydig cell here, decreased intratesticular testosterone level. This is the virus that so many virus also has an activity in male reproductive transmission. How about the DAP of the DAP full of COVID-19 or virus of SARS-CoV in male reproductive problems? Because of some shows that there is no present in semen, but and the others researchers shows that there is a presence of in semen. This is the part of investigation that find that SARS-CoV find in semen, including in Indonesia also. It's also in the patient with pneumonia, and then also in recovery problem, recovery state or recovery condition after COVID-19. Now, for the histopathology and ultrastructure of COVID-19 infection in testicle, it's so many damage of the testicle tissues. This is the normally, and this is after the COVID attack here. And then we looked at there's a decreasing or the damage, damage of the testicle tissue. Now, beside on the spermatogenesis, it's also potential effect of male gonadal function, especially fertility. The semen parameter in men recover from COVID, 90%, 60 until 90% shows that there's decreasing of sperm quality, decreasing of sperm parameters after recover from the COVID. What the problem? It's coming from DNA fragmentation and effect of the treatment. Yeah, the suspicious is from the effect of the antivirus here and the long COVID complication. Yeah. Now, other problems. What are other problems? The produce of antibody anti-sperm, and then nuclear damage of sperm DNA, and increasing of aneuploidy probability, and fibrotization of tissue, testicle tissue. Yeah. The problem also in laboratory management. Yeah, in sperm laboratory management. Vaccine is the very doubtful, but after there is a research shows that the vaccine is safe for the sperm. Yeah, it's okay for the vaccine condition. Now, what about in sexuality? COVID give a sexual problem, erectile dysfunction, because of there is a decrease, there is a damage of the endothelial cells in the corpus cavernosum. And then also desire disorder. There is the decreasing of testosterone and lead to the desire disorder. Yeah. And also in psychogenic because of from home and done during sexual desire problems, disorders, or disorders. Okay, infection or the inflammation in prostate, it's increasing of the probability, yeah, the worst condition of prostate cancer. Yeah, this is the new statement or new research that give attention in prostate cancer. Okay, last for the conclusion, the impact of COVID-19 is spermatogenesis disturbance due to the infertility, yeah, and sexual dysfunction problems, decreasing of testosterone, and others inflammation. It will lead to and it has potential effect on quality of life. I think my presentation is enough. Thank you for your attention. Assalamualaikum.
Okay, thank you very much, Dr. Dicky, for your interesting presentation. I think at the last, you have, like a problem in your connection or not? Or maybe my connection. Hopefully only my connection. Okay, we only have a limited time for discussion. So I welcome you to address your question to Dr. Dicky. This is interesting because people sometimes are always think about the respiratory tract, the cardiovascular system, and hematology system if we are talking about COVID. But Dr. Dicky is interested in exploring the effect on male reproductive organ. Okay, that's great. This is from Rizky and from Dr. Mila Hayati. Okay. Okay, should I read it for you? Okay, this is from Mila Hayati. Mila Hayati first, I think. Or, okay, Rizky first. Okay, Rizky. Okay, which of the many variants of COVID-19 that can significantly reduce male reproduction? Okay, what is the, the variant? There is any certain variant?
Yeah, thank you for the question. There is no research that shows about the safety on the variant. There's no research the male reproductive problems based on the variant. But I think for the research in the last I presented here, it's under the alpha and beta, alpha and delta, delta for variants. Yeah, because of delta is more horrible variants of the COVID. So many virus. But for the first time of case reports, there's alpha variants, so that there is a case report, the patient with the swollen testis. It's also due to the COVID, the variant of alpha. But there is no research that clearly to differentiate between alpha, beta, and delta or omicron. Thank you.
Okay, thank you, Dr. Dicky. There is a next question. If the COVID virus is found in the semen, does that mean it can be transmitted to a partner during sexual intercourse?
Yeah, I think, of course, but there's no data about this transmission. But learning from the others viral disease, of course, the viral transmission is via the sexual intercourse. It's probably clear virus transmission during the sexual intercourse. But in COVID, there is no data. There's it's clearly about the HIV and the hepatitis B. Yeah, it's clearly CMV. Yeah, it's via the sexual intercourse transmission. Thank you. Sorry.
Okay, thank you. Next one. Do anti-sperm antibodies appear due to the long infection of the virus or the effect of therapy?
It's an interesting question. There is no research about the produce of anti-sperm antibody. Yeah, there's no research. Yeah, there's no report from the researchers about the anti-sperm antibody. It's not just like in parotitis, yeah, from the mumps virus. It's clearly that the effect is a produce of anti-sperm antibody. But this is the chance for the researchers to show that the effect of the COVID also to make a production of anti-sperm antibody increase. Thank you for the information.
Okay, thank you, Dr. Dicky. There is still a question for you. Okay, will the pathological traits found in this man will be inherited by the offspring?
I think it's the worst probability is if there is attack in DNA of the sperm, yeah, because of due to the sperm aneuploidy and afraid to the production of the worst or the damaged quality of embryo. But it's still a question, yeah, because of there's no report on the patient. But I think so many patients, there is a horrible decreasing of sperm, yeah, after sperm. It's after the falling at the bottom, yeah, because of after maybe 80 millions per ml, yeah, per ml. But and after the recovery of COVID, it dropped until 0.9 until only 1 million ml, yeah, of the sperm. Yeah, very far. It's not about the effect on DNA, but in a number of the sperm production. Thank you.
Okay, thank you. There is another question. Dr. Diki, do you have any information on how wild animals or the virus carrier male reproduction system adversely affected by the virus?
Sorry?
How the wild, do you have any information on how wild animals or the virus carrier male reproductive wild animals or the carrier virus carrier its male reproductive system adversely affected by the virus? Do you have any information? Wild animals?
No. No.
No, because you are dealing with the human, right? Yeah, maybe we can share from the veterinarians. I don't have any information as well. Okay. Okay, another one, the last one, I think, I hope. Yeah. I would ask you about the effect of COVID-19 on male fertility, temporary or permanently. Thank you.
Okay, thank you. It's still under the research, and it's still under the assumption that the virus will be limited disease, yeah, and will be recovers automatically after 3 until 6 months. And so COVID is considered as temporary. But wondering is there is an increasing of worrying because of long COVID. It's due to the long COVID problems, and so many patients after 6 months recovery from the COVID still have symptoms of COVID-19 disease and COVID-19, and so it is carefully to make a conclusion, or temporary or not, yeah, or permanently. But we still know that after observation in the short period, there's a recover from the male reproductive problems after 3 until 6 months. Dr. Hera, thank you.
Okay, thank you. I think all the questions have already answered by Dr. Diki. And because I think the time is up for the plenary session for this today, or the first day. So again, thank you very much to Dr. Diki. Hey, where is the clapping hands? Okay, thank you. Before I close, I would like to say thanks again for all the invited speakers, all the participants who have, active participation in this session. And I hand in the agenda to Karisma, our Master of Ceremony.
Dr. Hera, thank you very much for your great session with Dr. Vijay, Professor Muranaga, and Dr. Diki. So to remember this event nowadays, ladies and gentlemen, we'd like to invite you to turn on your camera. But first, let me invite Dr. Hera, Dr. Diki, Pak Slamet, and Dr. Mona to join us in the first session of photo session, or virtual photo session. Yes, member who attend the symposium to join us in this online photo session. On my count, ladies and gentlemen, the first one, the IPS member, as well as the moderator and the invited speakers. So, in 3, 2, 1, cheers. One more time, ladies and gentlemen. In 3, 2, 1, cheers. Thank you for the IPS members and the moderator as well as the invited speakers. Now let's move on to the participants. We'd like to see you in this event, so we'd like to invite you to turn on the camera. Are you ready, dear committee? Okay, here we go. In 3, 2, 1, cheers. One more time. In 3, 2, 1, cheers. Another photo session. Let me count in bahasa Indonesia, Bapak Ibu. 1, 2, 3. That's it. Thank you very much for your participations in this plenary session. Don't forget, right after this, we are going to have the parallel session at 13 waktu Indonesia bagian Barat. So we'd like to have you here at the main hall or at the main room at the latest by 12:50 atau pukul 12.50 Bapak Ibu, untuk nanti bisa terbagi ke breakout room masing-masing. For the breakout rooms, we've got 4 breakout rooms. All of the presenters are already assigned parallel session. So make sure that you already fill in 2 attendance form, the plenary one and later at the parallel session. Alright, so after this break session, ladies and gentlemen, enjoy your discussion at the breakout rooms, and for tomorrow events, we'd like to invite you to join us at 8 a.m. with the same link, with the same password as well. And with this, I'd like to thank you everyone who already participated in the 3rd ISGP. And on behalf of the committee, have a wonderful discussion, everyone. My name is Karisma Devi. Thank you. Bye.
The Faculty of Veterinary Medicine of Universitas Gadjah Mada was established in 1946. It was started from the Bogor Veterinary Medicine College. In 1947, a parallel class was opened in Klaten. Faculty of Veterinary Medicine has 4 programs: Veterinary Medicine Study Program, Veterinary Profession Education Study Program, Master of Veterinary Science Study Program, and Doctor of Veterinary Science Study Program. Various tasks and expertises of veterinarian are learned in 12 departments. Each department is equipped with advanced laboratories and modern research tools. The 12 departments are Department of Anatomy, Department of Biochemistry, Department of Pharmacology, Department of Physiology, Department of Surgery and Radiology, Department of Internal Medicine, Department of Veterinary Public Health, Department of Microbiology, Department of Parasitology, Department of Pathology, Department of Clinical Pathology, Department of Reproduction, Obstetrics and Gynecology. Besides an advanced laboratories inside all of the departments, this faculty is also supported with animal health education and training unit, used by university students and veterinarians for the sustainable learning activities. This unit is called UP2KH, which manage several livestock such as cows, sheep, goats, and horse. Various achievements have been obtained by the Solari Society, such as the Regan Tester Kit for chicken carcass. FVM lecturers discovered this tester kit. It has been patented under the name Durante and has been widely used by the community. Faculty of Veterinary Medicine lecturers regularly write and publish books related to the veterinary medicine world that can be used for readers in the animal husbandry, biology, and statistics. The university students has also obtained several achievements in the research, arts, and sports field, and also national and international organizations. Some collaboration in the national level has been made. FVM UGM is the member of Indonesia Veterinary School Association. An MOU has also been signed between the government and private sectors for the purpose of research and community service. As for the international level, FVM UGM has also collaborations with foreign universities from Australia, Thailand, and Japan for research and education and other abroad universities. Furthermore, FVM UGM accepts international students from abroad, such as from Malaysia. The commitment to develop science and community service is based on the veterinarian work principles, Manusia Mriga Satwa Sewaka. We are to serve mankind through animal kingdom.
Universitas Gadjah Mada or UGM is a public university renowned as the People's University, which strives for and puts forward the interests of the citizens and nation of Indonesia. As a higher education institution that has long been recognized by the society, we are proud of our timeless and tireless dedication to the nation. By cultivating, implementing, and leveraging education, research, and community services, which constitute the salient objectives of Indonesian higher education or Tridharma, we remain committed to incessantly promoting the values of Pancasila. We exert the utmost effort to protect Indonesia's sovereignty in accordance with Pancasila ideology and the 1945 Constitution. As a world-class university, UGM is devoted to the interests of the nation and humanity. We conduct research, provide education, and deliver community services by enhancing knowledge that benefits the community, the nation, and the world. We have been learning from one another and get stronger together since we believe that knowledge is not useful unless it is shared with others. The learning process will never cease developing. At UGM, we learn by continuously exploring sciences, learning from history, observing the present, and researching for the future. We always work hard and never give up. The learning and research processes at UGM continue to run together and explore state-of-the-art innovations. To create a convenient, environmentally friendly, and pollution-free green campus, UGM has built a wisdom park that substantiates the teaching and learning process. The park per se is open to the public. UGM has also developed drinking water facilities throughout the campus, provided by Drinking Water Supporting Unit. This system is harnessed by the entire academic community of UGM. As the manifestation of its teaching endeavor, UGM operates various field laboratories for educational and research purposes. Furthermore, UGM has established and run a science technopark as an embodiment of its capability of inventing and commercializing a myriad of innovative health products, ranging from medical devices, herbal medicines, and healthy food.
There are now more than 118,000 cases in 114 countries, and 4,291 people have lost their lives.
2020 has been a tough year for anyone, and it is no exception for UGM. Campus activities are abruptly halted, student activities on campus are annulled, and everyone is forced to work, worship, and study from home. Lecturers have no choice but to learn how to teach effectively using the information and communication technology or ICT. Learning and research on technology are strongly supported at UGM, and in fact, we have conducted various cutting-edge technological research. Community Service or KKN is one of the most important courses to complete the study program, which will be undergone by University of Gadjah Mada or UGM in 2020 while adapting online courses system. Supported by the Ministry of Education, UGM will continue its service within the society across Indonesia during the pandemic issue. Lecturers and researchers at UGM continue carrying out basic and applied research in many realms. For instance, studies in the health sector to cope with COVID-19 are being encouraged and accelerated as a form of UGM's commitment to mitigating disaster using scientific methods. One of the most innovative inventions regarding to mitigating the COVID-19 is Genos, a screening tool or early detector of patients exposed to COVID-19, which is able to identify the symptom through exhale within 80 seconds. Genos is highly reliable. It has a number of advantages as a COVID-19 fast detection tool. Its sensor is able to detect more than thousands of patients in the long term, while providing relatively fast results, non-invasive and affordable. In addition, its usage also applies non-rebreathing mask and disposable HEPA filter. Those challenges somehow direct or even force us to innovate and modify approaches. Success is a journey which necessitates smart work through learning, studying, and sacrificing. Despite all that, most of all, we love what we do.
The Faculty of Veterinary Medicine of Universitas Gadjah Mada was established in 1946. It was started from the Bogor Veterinary Medicine College. In 1947, a parallel class was opened in Klaten. Faculty of Veterinary Medicine has 4 programs: Veterinary Medicine Study Program, Veterinary Profession Education Study Program, Master of Veterinary Science Study Program, and Doctor of Veterinary Science Study Program. Various tasks and expertises of veterinarian are learned in 12 departments. Each department is equipped with advanced laboratories and modern research tools. The 12 departments are Department of Anatomy, Department of Biochemistry, Department of Pharmacology, Department of Physiology, Department of Surgery and Radiology, Department of Internal Medicine, Department of Veterinary Public Health, Department of Microbiology, Department of Parasitology, Department of Pathology, Department of Clinical Pathology, Department of Reproduction, Obstetrics and Gynecology. Besides an advanced laboratories inside all of the departments, this faculty is also supported with animal health education and training unit, used by university students and veterinarians for the sustainable learning activities. This unit is called UP2KH, which manage several livestock such as cows, sheep, goats, and horse. Various achievements have been obtained by the Solari Society, such as the Regan Tester Kit for chicken carcass. FVM lecturers discovered this tester kit. It has been patented under the name Durante and has been widely used by the community. Faculty of Veterinary Medicine lecturers regularly write and publish books related to the veterinary medicine world that can be used for readers in the animal husbandry, biology, and statistics. The university students has also obtained several achievements in the research, arts, and sports field, and also national and international organizations. Some collaboration in the national level has been made. FVM UGM is the member of Indonesia Veterinary School Association. An MOU has also been signed between the government and private sectors for the purpose of research and community service. As for the international level, FVM UGM has also collaborations with foreign universities from Australia, Thailand, and Japan for research and education and other abroad universities. Furthermore, FVM UGM accepts international students from abroad, such as from Malaysia. The commitment to develop science and community service is based on the veterinarian work principles, Manusia Mriga Satwa Sewaka. We are to serve mankind through animal kingdom.
Universitas Gadjah Mada or UGM is a public university renowned as the People's University, which strives for and puts forward the interests of the citizens and nation of Indonesia. As a higher education institution that has long been recognized by the society, we are proud of our timeless and tireless dedication to the nation. By cultivating, implementing, and leveraging education, research, and community services, which constitute the salient objectives of Indonesian higher education, or Tridharma, we remain committed to incessantly promoting the values of Pancasila. We exert the utmost effort to protect Indonesia's sovereignty in accordance with Pancasila ideology and the 1945 Constitution. As a world-class university, UGM is devoted to the interests of the nation and humanity. We conduct research, provide education, and deliver community services by enhancing knowledge that benefits the community, the nation, and the world. We have been learning from one another and get stronger together since we believe that knowledge is not useful unless it is shared with others. The learning process will never cease developing. At UGM, we learn by continuously exploring sciences, learning from history, observing the present, and researching for the future. We always work hard and never give up. The learning and research processes at UGM continue to run together and explore state-of-the-art innovations. To create a convenient, environmentally friendly, and pollution-free green campus, UGM has built a wisdom park that substantiates the teaching and learning process. The park per se is open to the public. UGM has also developed drinking water facilities throughout the campus, provided by a drinking water supporting unit. This system is harnessed by the entire academic community of UGM. As the manifestation of its teaching endeavor, UGM operates various field laboratories for educational and research purposes. Furthermore, UGM has established and run a science technopark as an embodiment of its capability of inventing and commercializing a myriad of innovative health products, ranging from medical devices, herbal medicines, and healthy food.
There are now more than 118,000 cases in 114 countries, and 4,291 people have lost their lives.
2020 has been a tough year for anyone, and it is no exception.